Everyone. We are starting the seminar. It's a seminar quite full of computational modeling and approaches, which makes it really appealing to the session. So I hope it goes smoothly and we can enjoy it. But we are starting straight away so we don't miss the last part of the seminar. We need to be precise. But please do ask as many questions as you can and interrupt by putting the questions in the chat, and we will monitor through William, who is a co-host, and me, and we'll read them aloud. Also, if you want to read them aloud, you can come into the scene and say the question aloud. So the first speaker, it's Dr. Duncan McGregor from University of Edinburgh. He's a senior postdoc in the Centre for Discovery of Brain Sciences, and he will talk us about the role of the hypothalamus in decision-making behavior with some computational modeling. So Duncan, whenever you are ready, off to you.
Okay. Hi all, it's great to be here at Neuromatch, having a brilliant time, all these amazing talks. So this, if you ignore the sort of cumbersome title, is basically about using modeling to predict network structure from single neuron recordings. So basically looking at the patterning single neuron recordings and applying the model, try and fit them and make guesses what the actual network is. So this is work in the VMN and sort of the ventral medial nucleus of the hypothalamus. Conventionally, it's involved in appetite regulation, but it seems to be involved in various other behavioral things. And we think it's probably a sort of decision-making center. So this picture, this green picture here, is showing you roughly where the ventral medial nucleus is. This is the third ventricle and it's the hypothalamus, and you can see these sort of green blobs of the cells. But from this, we're going to get away from the real looking biology and get into sort of abstract things. So ventral medial nucleus, regulator of feeding, probably also sexual behavior, also seems to involve in aggressive behaviors or fight or flight behaviors. It does a lot of things to do with energy homeostasis. So it's involved in glucose homeostasis, thermogenesis, as well as appetite regulation, sends signals to the other parts of the hypothalamus. They're well known at being involved in appetite regulation, make decisions whether to eat or not. It's got various input signals which you expect to be involved in this. There's sort of peptide signal ghrelin, which is usually comes from your stomach as empty. There's leptin, that sort of signals your fat reserves, another hormone signal, glucose. Oxytocin, which is another sort of quite well-known peptide signal released in the hypothalamus, is involved in lots of things, oxytocin, but has a big role in appetite regulation. We know it's got output signals that control food intake in various pathways. It also sends controls to places involved in glucose homeostasis. It might send things even to the liver, direct signals. That's kind of vaguely known at the moment. Also energy expenditure things. So the thermogenesis is one of the sort of big ways in which we can make a control over energy expenditure. It's probably a decision-making center, we think. It's coordinating a lot of different behaviors, which are ones which tend to be exclusive of each other. So you'd have to decide whether to sort of favor one or the other. So it probably is making decisions between these, but certainly it's a center which is doing sort of signal integration and is sending sort of strong signals, or involved in deciding which behaviors to activate somewhere. Most of the data we have and understanding what it's doing is from sort of lesioning studies. So if you lesion the VMN, then animals get very sort of obese. Lots of molecular studies looking at the sort of signals and receptors that are there. What we really want to do is try and get a better understanding of how the actual electrical activity, how the neurons are actually processing these signals and what they're doing. So basically we did lots of recording of the neurons. This is work happened about 10 years ago. I'm in a lab which is primarily experimental. I do modeling there, so really great sort of direct access to lots of electrophysiology data. So basically it's just recording in vivo from lots of VMN neurons. This is single neuron recordings, extracellular in anesthetized animals, and basically process all these recordings by extracting the interspike intervals. So basically you have all your spike times, you count the actual interval between these spike times, and you make histograms of these, and you can process these histograms into these things called hazard functions, and then apply lots of statistical measures of, you're just looking at the firing rate, sort of variation in firing rate, various measures of that, and basically use these sort of statistical measures just to sort of divide these cells based on their patterning into sort of types or potential subpopulations of cells. And you can see here, this is sort of rough idea of all these types, and they're basically just named on sort of descriptive things of all their actual features in their distributions, which I'm going to show you. So if you focus on these, this is the sort of spike rate. This is spike rate binned over 1 second. And this gives you a sort of rough idea of the activity in a cell. It basically just, you can see how noisy the activity is and get a rough idea of the sort of spiking rate. And if you look down, these are different types of cells which are classified. There's not a lot you can see in the sort of spike rate. And you can see where some are faster than others and some are noisier than others. So obviously different one here is what we call the regular cells, where it's actually very regular spike rate, very sort of regular intervals that don't change very much. This random cell, this is quite a sort of typical sort of histogram. You've seen a lot of different types of neurons in the hypothalamus and other parts of the brain. So basically, if you just have random inputs to a cell, a purely random cell will basically give you sort of exponentially decaying distribution. It's basically a sort of Poisson distribution. But most cells have got at least a missing part at the front of this histogram where you have a hyperpolarization that follows a spike, which then gives you a refractory period. So you're basically missing a chunk of the short spike intervals. And then as we go down these different types of cells, we have different sort of features. This, what we originally called a slow DAP, it really turned out to be a bit of a misnomer, but based on other cells that were explained this way, you have a bulk of very sort of faster intervals at the front of the histogram, and then sort of normal, more sort of random things. And then we have these long tail cells, which has this very particularly long refractory period. So this is 100 milliseconds here, so that's quite, that's particularly long compared to other neurons we've seen. And there are long tail too, it's an even longer refractory period. And then this broad cell, which is a sort of a very wide sort of histogram. And we also apply this analysis called index of dispersion. So index of dispersion, each of these bars here measures the variability. So basically this is a sort of way of quantifying noisiness you can see in sort of spike rate here. And we quantify it on different time scales. And this is a really useful way of measuring patterning on longer time scales. So basically, the takeaway from this is your interspike interval histogram gives you sort of short timescale patterning, and your index dispersion range gives you longer timescale patterning. And you can use these to define and then fit the cells. These cells over here, so these ones are variations of sort of random cells at different refractory periods, and nothing too sort of complex and things we often see. These cells over here on the right are sort of stranger ones. So you have the regular cells, and then you have these doublet cells, and these are the doublet ones. Are basically the spiking is made up of very fast doublets and triplets of spikes. So, very short intervals, just very, very short bursts. So this big spike at the front histogram is the sort of intervals between these short bursts, and the rest is just the sort of intervals between all these like individual doublets and triplets. And then there's combinations of these. This doublet broad is kind of a mix between sort of what we saw the features of the broad cell and the doublet cell. So it's got a mixture of very sort of long refractory period and also these little short bursts. Then the strangest, most distinct cell is these, what we call oscillatory cells. So they've got multiple peaks within their histogram, and that indicates some sort of underlying rhythm that's driving excitability of these cells. And our idea, which we sort of developed in the model, is that these are some sort of network features are generating rhythm, and this combined with sort of random input to produce this oscillatory peaks. So integrating FAR-based model is what we've used. I mean, basically it's a very sort of quite a simple spiking model, which we then augment by adding sort of hyperpolarizing potentials, these post-spike potentials, and making different combinations of these. So what we call the HAP is a sort of very large magnitude, fast decaying hyperpolarization. The DAP is a depolarization, sort of not as fast decaying, and the AHP is similar to the HAP, but it's a sort of smaller magnitude, but longer lasting. And there's no reset in this version of the sort of integrating FAR model. So basically these post-spike potentials can summate across spikes. And then we can apply these. We used automated fitting using Hierarchical algorithm to basically match the activity in these cells. Basically, you can just take a few parameters, the sort of input rate, the magnitudes and time course parameters, the HAP and DAP, etc. And you can get these really good fits, these really close fits to the spike histograms and also hazard functions. So you can see here, we've got all these quite close matches to these different types of cells. And this then gives us a prediction of what are the intrinsic features that are producing these patterning. And the sort of dominant thing that varies between these is the time course of the HAP. So some of these use quite short HAPs, and some use like longer ones to use, for instance, like the long tail effects, these long refractory periods. we also have these matches to the index dispersion range, which helps pin down things like the AHP and the DAP. So the other types of cells we couldn't fit with a single neuron model. And at this point, the simplest explanation, sort of Occam's razor, becomes that it is actually a network feature. And we do know that these cells in VMN, there's not much evidence of inhibitory interconnections, but there is a lot of evidence of excitatory connections in these cells. There's a lot of glutamatergic synapses. So we basically took our fitted integrated fire model or single neuron model, and we added a very simple model of excitatory synaptic input between these neurons, made a very simple network. So if we're taking a sort of fit to one of our long tail cells, basically what we're doing as we go down here is we're increasing the density of the synaptic connections. As you increase the density, you start to see the other types of cells emerging from this. So then you move to sort of long tail 1 cell, so this refractory periods become shorter. We keep increasing them still, and this peak, the second peak in the histogram appears, and then we get what's like doublet broad cells. And then if we increase the connection density even more, we start to see basically matches to our sort of doublet cells. And one of the interesting things that get produced in this, if you look at the summed activity over a population of these cells, you start to see these oscillatory sort of patterns. So in a single cell, you have these sort of short, basically these increasingly regular bursts. But looking at population with a bit of noise in the connections, you start to see these almost oscillatory signals. So that was basically, a possible source of the oscillatory signal we saw in the actual oscillatory cells. So we moved on to sort of testing that in another network. One of the other things that came out of this, if we tried, was making a network of different type of cells. So the previous one was just a slow HAP in this neuron. If we add the DAP, which is seen in other neurons, to this network and tested what it did, we saw this basically bistability. So we had actually seen in the recording some cells that had sort of spontaneous bistability. They would switch between fast and slow firing over quite a long time scale. This example at the top is from the in vivo neuron. And we basically put this in the model, and it dropped out of the very model very easily. There's no explicit bistability in this model. It's purely a sort of emergent effect where we just have the combination of the inhibitory effect of the HAP combined with the excitatory positive feedback of the connections and network, and also the DAP itself. And then we can very simply get a match to this bistable activity, and we can simulate in the model things like inhibitory signals. This CTK is basically a classic gut signal which affects a lot of the hormones, the neurons which are involved in appetite regulation. So we basically just simulated this with a short inhibition to the synaptic input to the model, and this basically drops off and then eventually goes back up to this fast firing state. And we can go further with this by combining excitatory and inhibitory signals. So basically now we've got a network where we can just basically switch on or switch off with a transitory signal, and it will sustain in this high activity state until you give it another intense signal to switch off again. So this is a potential mechanism for decision making. The final interesting network I'm going to show you is how we managed to match the oscillatory neurons. So we couldn't match this with a single cell type network, but we realized we could produce the underlying oscillation that could drive these peaks from a slow HAP network and then feed this into faster HAP cells. So basically we need faster HAP cells so they can get this peak at the front of this histogram. You can't get that if you have a long refractory period. So basically you're using the slow HAP cells to generate an input, an oscillating input signal to another type of cell or fast HAP cells. So the slow HAP ones, their histogram looks like this. And the fast HAP cells are getting this oscillating input, start to see this. We get a very close match to what we'd observed in the recorded cells. And we can kind of see how it's working from looking at the summed activity, the slow HAP ones. We see this sort of oscillating signal. Now, to get this to work and to match the histograms we've seen in the recorded neurons, we had to put noise into the connections in the network. So if you have...
Sorry, Duncan. There is 1 minute ahead of time, yeah. But
yeah,
after the time, yeah.
Okay, so anyway, basically, slow HAP1s are generating this with a bit of noise in the connections by varying the latency of the connections, the varying delay between the synaptic transmission. Then you get this nice oscillatory waveform, and this produces this oscillatory signal we see in the neurons. And that is that. Thank you for listening.
Thank you, Duncan. Is there any question? There is no questions. Just a quick one I would like to comment. So there is all these models and external, internal inputs, but how do you associate it to the decision making itself? I'm not familiar with this type of topic, but where do you translate into decision making?
Well, that's basically, we're basing that. I mean, we think that the VMN is involved in decision making because it seems to arbitrate between these sort of behaviors that can't coexist. Basically, what we think, I mean, we haven't got all the way to an actual decision making mechanism, but we think this bistability where you can switch on and off a signal in a sort of conditional way is, that's a basic mechanism that could be involved in decision making. You've got an all or nothing signal which you can robustly control.
Okay, you don't do actual behavior after these models. Okay, I was just my own bias, I guess.
This is as far as it's got.
Right. Okay, I think we have to move on. Otherwise, we get kicked out of the room. So, thank you, Duncan. While Amadeus, can you start sharing your screen? So the next speaker is Amadeus Maes. He's a PhD student at Imperial College in London, and he will tell us about learning sequences with multiple timescales and through a spike in neural modeling. So it's quite a related topic with the previous talk. So whenever you're ready, Amadeus.
Can you see my screen?
Yeah.
Okay. So good afternoon. I will talk about learning compositional sequences using a hierarchical network. And so what is compositional behavior? Compositional behavior has some vocabulary, multiple motives or elements are combined together to form the behavior. And one example of this is, for example, birdsong. Here at the bottom, you can see a spectrogram of a Bengalese finch, and you see that in the spectrogram, there are motifs that reoccur during the song, and the ordering somehow produces the behavior. Okay, so there are two main ideas of how such compositional behavior is implemented in neural systems. There is a serial model versus the hierarchical model. And for this, let's consider an example sequence, the sequence here, A1.
We cannot hear you, Amadeus.
He seems to have become unstable. Let's give him a minute.
Okay. Amadeus?
Yeah, sorry, I got disconnected for some reason. Yeah. So let me share again.
Start.
Okay, so where was I? I was talking about the neural implementation of compositional behavior. And so in the hierarchical implementation, we have elements A1 and A2 that are grouped into motive A, and elements B1 and B2 that are
Amadeus,
grouped into...
Yeah. We don't see your screen.
Okay.
Yeah.
And now?
Yes.
Okay, sorry about that.
No problem.
But now my camera is off. Okay, so there's an increasing experimental evidence for hierarchical structures in multiple animals, as shown in these papers. And there are also good theoretical arguments for a hierarchical model, and I will talk specifically about three things today. We'll talk about flexibility, efficiency, and robustness. And so in the model I will propose, the main ingredient is sequential dynamics. Sequential dynamics has been recorded in multiple experimental papers, for example, in birdsong, but also in navigation and in decision making. And in theoretical work, it has already been shown in multiple papers that sequential dynamics can be learned in spiking neural networks using local plasticity rules, and that it is beneficial for learning dynamics in readout populations. So let's say that we want to learn a target sequence AAB. So you can see that this motif A looks like this, motif B looks like this, and this target sequence is 1 second long. And this is the serial model that I will propose. So this serial, we have a serial clock here at the left, which projects to these readout networks on the right, and each readout network encodes a motif. In the serial clock, we have excitatory clusters. So there is a sequential dynamics here because each excitatory cluster activates the next excitatory cluster and so on. And when you repeatedly present this target sequence to the readout networks, these readout synapses are potentiated and the target sequence is stored. So you can see this, for example, in the raster plot here, where at the top you see the sequential dynamics of the serial clock, and at the bottom you see the dynamics of the readout populations, and you see that indeed this target sequence AAB is replayed. And to verify that indeed the target sequence is stored, you can look at the readout weights, and you can see that the target sequence is nicely stored. So how do we now move to a hierarchical model? In a hierarchical model, there are, we propose that there are sequences on different timescales. So instead of a single recurrent network producing sequential dynamics, there are here two recurrent networks, the fast clock here and the slow clock at the bottom. The fast clock displays sequential dynamics on a timescale of the individual motifs, and the slow clock produces sequential dynamics on the timescale of the entire behavior. So the fast clock is directly connected to the readout networks through these motif synapses, which encode the motif dynamics. The slow clock, however, is not directly connected to these readout networks, but it is connected through this set of interneurons. And this set of interneurons, you can essentially interpret them as a control module. It controls which of the readout networks is active at any given time. So because of this, the syntax synapses can store the ordering of the motifs. The both pathways are plastic, and they follow this simple local SCDP rule with the window shown in this cartoon. Okay, so can we now learn the same target sequence as before in this hierarchical network? So let's say we present this again repeatedly to the readout networks. Then this is what you get. This is these spike rasters show spontaneous dynamics. So you can see here the fast clock displaying fast sequences, and the slow clock the long slow sequence. You can see that in the readout networks, the target sequence is replayed. You can see AAB, and then you see that the interneurons determine the order of the dynamics of the motifs in the readout networks. You can now look at these the synapses in those two pathways. In the direct pathway and those motifs, the motif synapses encode two motifs. And this indirect pathway, the syntax synapses, encodes the ordering on the longer time scale. Okay, so now we wonder how flexible is this? Can we, for example, relearn from when we first learn AAB? Can we then switch the two last motives and can we learn ABA? For this, we look at the error and we can define the error as the difference between the spontaneous output of the network and the target sequence. First, let's look at the motive error. The motive error decreases independent of which sequence is presented. So the first 50 iterations AAB is presented, and then the next 110 iterations ABA is presented. The motive ordering error, however, does change dependent on which sequence is presented, of course, where we see the brown line, which is the error with respect to target sequence AAB, and the green line is the error with respect to the second target sequence. Then we can also look at the total sequence error, and you can interpret this total sequence error as the sum of the motive and the motive ordering error, and you see that there's an almost discrete switch where at a point AAB is unlearned and ABA is learned. Now we can compare this behavior, this learning behavior, with a serial model, and this you can see in the right panel here. And you can see that in the serial model, the relearning is much slower, it's much more gradual. And this is, of course, because there's only a single pathway and because both the content of the motive and the ordering of the motive has to be relearned. Okay, so let's move to now learning multiple sequences. Let's say we want to learn both AAB and the second sequence BAAB. In a hierarchical model, here at the left, you can see that all that is needed is an additional set of interneurons that encodes the syntax of the second sequence. And you can still give supervisor input to the same set of readout populations. However, in the serial model, there is only a single pathway, and this is not possible. So you have to add readout networks for each new sequence that you want to learn. And so there is a certain inefficiency related to the serial model. Finally, I'll talk about perturbations. An example perturbation that I will show is what happens when you perturb the serial clock. So in the spike raster here in the right, you can see that the sequential dynamics in the serial clock breaks down. And because this breaks down, also in the readout populations, there is no more activity. So the first motive is played, but then the second and the third later motives are not played. In the hierarchical model, however, this is not necessarily the case. Let's look at this specific perturbation here. So we perturb the fast clock here. You can see this in the spike raster that after the first motive is played, the second motive is deleted because of the perturbation, but the final motive is still played because of the existence of the hierarchy. Okay, so to summarize, I talked about hierarchical model being more flexible, more efficient, more robust. And for more details and more simulations, I would like to refer you to the bioRxiv paper. And I would say that future work can include making more quantitative links to specific behavior, specific brain areas. And one thing that we're working on right now is to see to what extent we can discover such multi-timescale structures in real data. And with that, I would like to thank my supervisors, and I'm happy to take questions.
Thank you, Amadeus. That was really cool. In fact, there is one question for you from Kirubel. Okay. I will read it aloud. He's saying, are they all LIF neurons? It's kind of
yes,
a second.
So these neurons are spiking neurons. They're actually exponential integrate and fire neurons.
Okay.
So there's typical.
The second point is, which STDP rule did you use?
Right, so so
because
this is
the problem he refers to is that he can't imagine this model being applied to language learning.
So is the question now, what this STDP rule is, or?
Yeah, yes.
So this STDP rule is simply a local rule. If two spikes are sufficiently close together, the synapse potentiate. So the time scale here is 5 milliseconds. When it comes to language, I think language is a little bit more complicated than this model. This model is very much about stereotypical behavior. It's more realistic, I think, for the example of birdsong, as opposed to human language.
Okay. Okay, thank you, Amadeus. We need to move to our last speaker. Thank you so much.
You.
So our last speaker is Christopher Lewis. He's a postdoc at University of Zurich. And the topic to finish this seminar, it's on the cortical resonance that has a selection role in coherent input. So Chris, whenever you want, off to you.
Great, thanks. Yeah, and thanks to the organizers and everybody for putting this together. So yeah, I'll talk about a resonance that occurs in cortex and how that resonance may affect transmission of signals through a cortical area. And most of this work was actually performed when I was at the Ernst Rungmann Institute in Frankfurt, Germany. But yeah, I'm now in the University of Zurich. And so rhythmic activity in the brain is ubiquitous. These are examples from Hans Berger of the first electrical recordings from the human brain. And there, one of the clear kind of, results from the data was that activity can be quite periodic. And indeed, here's an example trace where the subject had the eyes open up until when this arrow is presented, and then they close their eyes and you see a large amplification of a slow rhythm. So this is around 8 or 10 hertz. This is the cortical alpha rhythm. And subsequently, since then, oscillations have been found more or less all over the brain. And so they occur across quite a few temporal scales. So from very slow oscillations, even across longs of minutes. And so I think you can even think of circadian rhythms occurring across days or longer. And they also occur on very fast scales. So up to what's called like gamma band oscillations, which are in the range of kind of 30 to 80 hertz or so, and even faster than that for muscle-related oscillations, or for example, like hippocampal ripple oscillations, which occur kind of in the range of tens of milliseconds. And they also occur across all species and brain structures. So it seems like it's a quite ubiquitous phenomenon. They've been correlated with different cognitive or behavioral variables. For example, if a mouse in this case traverses a linear corridor, in the hippocampus, there is a strong theta rhythm, and place cells in CA1 will spike preferentially for given spatial locations. And those spikes will occur at preferential phases of the theta oscillation with respect to the mouse's position and trajectory in space. These same sequences can occur at faster timescales. So about 10 times faster, for example, in the ripple band when the mouse is at rest. And these sequences can reflect previous trajectories the mouse has gone, or also future potential trajectories. In other wake behaving animals, for example, cats or monkeys, also humans, if they're monitoring a visual scene, in this case cats, and an expected object is presented to them, you see strong alpha band synchronization in visual cortex of these animals, actually visual and parietal cortex. And you see gamma band synchronization as well, but it's quite reduced as compared to a novel stimulus. So some sense of violated expectation can give rise to larger synchronization in the fast frequency range and reduced synchronization in the low frequency range. So there's I think a large body of evidence like this that correlate synchronization or oscillations in different frequency ranges with different cognitive or behavioral effects, but there's still a lot of disagreement in the field about what the actual role of these oscillations might be. I think it even goes to the extent that many people believe that they're simply epiphenomenal in a sense, so they don't have any kind of causal role in the system. They simply occur when you have coupled excitatory and inhibitory elements, and the oscillations per se don't actually facilitate any sort of information processing. So this is a question which is of interest to me because they are so pervasive. In some sense, Occam's razor might suggest they should play some role in the functioning of the system. And so one compelling proposal for the function of synchronization is actually in facilitating dynamically routing information in the brain, because depending on behavioral contingencies or goals, it can be the case that you don't want to simply propagate all information to subsequent layers in the cortical hierarchy, for example. So in vision, this is the case that you have a lot of potential sensory information, but higher order visual areas might not necessarily reflect all of that information, and information from the primary visual cortex might be selectively propagated according to its behavioral relevance. So you can imagine a monkey looking at some visual scene, and it might find two objects in that visual scene which are behaviorally relevant to that monkey. And these objects, that in this case the pear and the apple, will be represented topographically in area V1, but area V1 will then converge onto subsequent downstream visual areas that have larger receptive fields. So you end up with a situation in which the neurons in the downstream area have to, in some sense, decide which visual stimulus to represent, or alternatively, they could kind of be in a bistable state where they reflect some part of each. And so one proposal that has been put forward is that when you have something like attention, that highlights object of interest, and that object is then selectively propagated to downstream populations. And there's a considerable evidence that this, at least that this does occur in monkeys, for example, from single unit recordings in area MT or V4, that not all information gets through, that there is some sort of informational bottleneck or some sort of flexible routing of information. And one possible implementation of that is synchronization. So if a visual object is on the screen, presented to an animal, so in this case on a computer screen, the monkey is freely viewing, this is data from Nick Brunet, an orange, then you get strong synchronization. This is a recording from area V1, and you see strong gamma band synchronization. So this is synchronization around 80 or 90 hertz. And this gamma band synchronization, we know from previous in vitro work and also modeling work, arises from the interplay between excitation and inhibition, because you have strong feedback inhibition from, for example, pyramidal cells onto PV interneurons, and then the PV interneurons can strongly inhibit the pyramidal cells, which give rise to kind of periodic excitatory periods. So this is kind of displayed here. So if you have some object represented in V1, you can have periods where excitation increases kind of the representation, you could say, of that object. And then that, once it reaches a certain level, activates the inhibitory network, which then suppresses spiking in the population. And then because inhibition has a time scale of some 10 milliseconds or tens of milliseconds, 20 milliseconds, then you get a rhythmic excitatory period, which then recurs in time. So if you have two visual stimuli, then you would have two different rhythms. And these rhythms would depend on physical characteristics of the stimuli, but also potential top-down signals, for example, behavioral context and so on and so forth. And then, so these two visual signals then converge onto a higher order. So for example, area V4. And the proposal, which was put forward by Pascal Fries, the communication through coherence hypothesis, is that the population, which is synchronized to the downstream neurons, will be preferentially transmitted. So this is actually then some mechanistic role for oscillations in the brain. But so far it's been hard to causally test this, because you can find selective synchronization, and there's been quite a bit of evidence that V1 and V4 are selectively synchronized, for example, under attention. But you never know if the oscillations are there simply because they're, for example, arousal or excitation in the network, or if they actually play a role in the selective routing. And so we wanted to try to test that hypothesis. And so In order to do that, we expressed channel rhodopsin in pyramidal cells of the cat visual cortex in area V1 and V4, a homologue of V4, which is area 21A. And we had expression really limited then to pyramidal cells. And this gave us control over the excitation in the local network in area V1 or in 21A. And so if we shine blue light on the cortex there in V1 or in 21A, in both areas essentially, we were able to induce strong gamma band synchronization. So this occurs around 40 to 80 hertz, depending on the strength of the excitation. But this gave us good experimental control over these oscillations. And if we look, this is the local field potential. And if we look at the spiking is very locked to the synchronous oscillation. So the pyramidal cells are basically becoming activated. This activates presumably feedback inhibition, which then gives rise to this gamma oscillation, which then subsequently locks the spiking of the neurons to the population rhythm. But we wanted to actually try to get causal evidence for flexible routing. And so in order to do that, we switched from using, this is a light which was static in time, so the light wasn't flickering, and the rhythm is purely intrinsically generated. And so to try to get a handle on causation in the system, we actually used white noise laser stimulation. So we flickered the laser with Gaussian distributed white noise with a mean intensity, which matched the constant illumination. And this temporally varying stimulation also gave rise to gamma band synchronization. But the advantage to this signal is because the white noise is completely uncorrelated in time, we can actually look at the effect of individual elements of input into the network on subsequent output of the network in terms of spikes in that network. And so to do this, we use the spike triggered average. And so what we do is we basically take every spike from units in the recorded population, and we align the elements of white noise to those spikes. And this allows us then to look to see if there were specific signatures in the white noise time course which gave rise preferentially to spiking. And in fact, we found that there was short bouts of gamma band input preferentially led to spiking in the network, suggesting that the resonance, the gamma band resonance of the cortical network actually gives rise to selective filtering of temporally varying input. So you can see that here. Also, instead of using a spike triggered average, we computed a statistical model between the white noise input and the action potentials of the population. And here you can see that really specifically in a relatively broad gamma band between, for example, 50 hertz and 120 hertz or so, we see the strongest effect of variance in the temporal input on spiking in the population. So it seems like the gamma band resonance is effectively selecting elements of the input which match its resonance. So to try to better understand this, we implemented some recurrent network models of excitatory inhibitory units, and these are simple point neuron but conductance-based Hodgkin-Huxley-like models. And if you give DC input to this network, it's well known that it generates strong gamma band synchronization. And so this has been studied by a number of groups, but we were specifically interested to see if the model would also reproduce the selective filtering effect we saw. And in that case, we actually then stimulate the excitatory units with white noise, and we see that there is selective transfer of the element of the white noise, which is coherent with the network resonance. And so it seems like this resonance does play some role also in the models in filtering it. And because we have the model, we can try to then understand the mechanisms of this. And so that's kind of what we do. We look at what is performing this. And so we isolate excitation from inhibition, and we only impose rhythmic inhibition onto a separate excitatory population, which is driven by white noise, which is independent. And here again, we find filtering. So it really suggests that the key of this dynamic or this filtering of the input is occurring through rhythmic inhibition. So it seems like rhythmic inhibition can basically preferentially transmit signals which match its preferred timescale. So that's the end of the story for here. And there's more I could talk about, but I think time's limited. And if you have questions, you can contact me at email or on Twitter. I'm on Twitter. And I think the people who worked on this with me, Jiangwang Ni and Thomas Wunderle and Patrick Indritza, all performed experiments with us. And Andrea Lazar helped with the modeling work, and Ilka Diester and Pascal Fries were involved in the supervision of the project. So
thank you. Thank you very much, Chris. It was very interesting, in fact. I see there are no questions so far, so while they come, because we have like a few more minutes, I'm interested in the causal approach that you use. You use a white noise light versus the blue light. What are the characteristics for this causality?
No, the light was always blue. It's just that it's temporal white noise. So because the blue light is what activates the channel rhodopsin, right? And yeah. So then it's basically we either have it with no temporal structure, so like a DC or a step function, which then is providing some constant excitation into the network, and that generates a kind of intrinsic gamma oscillation. Or we flicker the light with Gaussian distributed white noise. So then we have a mean value which matches our constant illumination, but we have every sample. So in this case, it was around 1 kilohertz. We were drawing a new intensity for the laser. Yeah, so the laser was just flickering essentially.
Because I guess all other causality methods that I know of, they are more invasive. Whereas this one is quite elegant in a way. You perturb the signal on the cortical region, but you let it function as it is, right? So it's quite clean as a control.
Yeah. I mean, so this has been used to understand single cells and things like this for like Panchklappen recordings. For example, I think Duncan kind of maybe alluded to this a little bit in his study, where you can patch a single cell and you can put white noise time sequences in. So people have used this to look at if cells have resonances, for example, or but then I think that applying this at the level of the intact system is very interesting. I think you could then study different circuits to see if they have different sort of transmission properties and stuff like that.
Okay. Since there are no more questions, I will have another. There are no more questions. I have another curiosity on this, more in the mechanistic part of the V1 and the V4 selection mechanism. Do you know, or do you get any gamma correspondence between both that can say there is a change between both regions in the object representation? Like, in other words, does the object from V1 to V4 have a somatotopic kind of transmission, or is it modified in the gamma that you see? I don't know if I was clear.
Yeah, I think so. There is evidence that synchronization is altered by, for example, like attention. So if you have two visual stimuli, I think the most standard, there are two experiments I can think of off the top of my head, one from Conrado Bosman, where he had two visual gratings, which were in different parts of the visual field. And so in V1, they were activating different patches of cortex, and those converged onto some V4 population, which was then measured. So the V4 population represented both spatial locations, but V1 differentially represented those different locations. And under a single case, so stimulation with a single grating, that grating would be represented in V1 and then also presumably represented in V4. And you could see this through synchronization between the two recording sites.
Okay. Right.
But if you had two stimuli in V1, the monkey, based on how its attention was oriented towards one grating or the other grating, which was done through a visual cue, would basically selectively synchronize with the attended object, and there was no synchronization with the non-attended. There's even a very elegant study of this also from Iris Grota from Andreas Kreiter's group. And there they also used white noise stimulation in the visual, but in vision. And so they basically had two patches on the screen, and the patches were flickering with different white noise time series. And so then you could see how attention selectively transmitted the flicker from the attended stimulus versus the non-attended stimulus.
Yeah. Okay. Cool. Well, we're gonna get kicked by the next session. So Thank you, everyone. Thank you, the speakers, for these nice and interesting topics.
With our panel, like the back end of the NeuroMatch, so I'd probably be taking a screenshot when you're on screen. I just wanted to let you know about that. Yeah.
Have we done that?
Oh, sorry, one second. I'm just trying to pin you. Let me stop share. Yeah, this is perfect.
Mm-hmm.
Got it. Thank you.
Thank you. Yeah, I should bring down my eyes, real eyes. These eyes are fake ones. Yeah. So actually, the thing inside this skin is the costume, and I'm really this green skin,
Oh,
dragon-like thing. Yeah. And this is my little friend.
okay. Let me take it again.
You know, the well-shred dragon.
You know what? Just keep the camera rolling.
Okay. All right. So I'm just waiting a few more seconds to let more people come in.
Sure.
Oops. Okay, so somehow Zoom hasn't let me capture a screenshot, which is weird. Okay, got it. Yes. I think we should get started now. All right. Okay. Yeah. So welcome everyone to the final day of NeuroMatch 3. Hope you have been having a great time so far. My name is Shankar Alagappan, and along with me, I have Furzan Farhani. Today, we have 3 speakers on an area that fascinates me a lot, computational modeling of memory using artificial neural networks. The format of the current session is an interactive talk, which means the speakers spend about 5 minutes introducing their topic, following which we have about 10 minutes for question and answers. We have the option of bringing you on screen to ask your question, or if you're very camera shy, I would recommend you use the Q&A panel available in Zoom, and also to vote for your favorite questions. And in the meantime, feel free to chat with the panelists or other attendees using the chat function. So without further ado, we have Ihar Liu from Cardiff University, who will be talking about memory performance in a network trained with generative replay. The floor is yours, Ihar.
Cool, let me share my screen. You can see it, right? Okay. So hello, everyone. I'm Ihar. I'm a postdoc from the NAPS lab in Cardiff University. Our lab focuses on sleep research. We believe sleep is crucial for memory, learning, task solving, and so on. And I study sleep with computational models. Here I'll be talking about memory replay in an artificial neural network. Given the time limit, I'll be skipping a lot of the details, so do leave any questions or comments. Okay. Yeah. So briefly speaking, there's a huge problem called catastrophic forgetting in artificial neural networks, which is not human-like at all. To overcome it, we use replay. Here, replay is used in a restricted Boltzmann machine. As a result, we see not only replays can prevent catastrophic forgetting, but also can replace with low quality. Note our model is very simplified, abstract, and high level. It is somewhat comparable to real humans, perhaps in terms of the neural population activities and the test performance, but not in all aspects. I'll skip the comparisons, and I'll skip all the mathematical and algorithmic details, but we can discuss them, and I do have these slides. First of all, what is an RBM or a restricted machine consists of these binary neurons, and there are 2 groups of them. The visible neurons see data directly, while the hidden ones don't. These weights between visible and hidden neurons are symmetric, and there are no connections within a group. One major property we like about an RBM is the generative ability, which essentially means the RBM is capable of predictive coding. After learning, it can generate patterns similar to training samples on its own. And in fact, even during learning, the learning rule it uses called contrastive divergence, it uses data from two different phases to update weights. The first phase is data-driven when the RBM sees the real data, and the second phase is model-driven when it generates some pseudo data. The RBM can contrast these two phases and updates its weights proportional to the differences. So RBM are very useful and have many applications, but like many other artificial neural networks, they suffer from catastrophic forgetting. The problem is basically referring to the extreme case of the stability plasticity dilemma. When the network starts to learn a new task, its memory traces of old tasks could be erased rapidly and entirely. There are different ways to deal with the problem, and we use memory replay, an idea following the theory of complementary learning systems. In our setup, the RBM needs to learn 10 classes of patterns, class by class, in an incremental way. So to avoid catastrophic forgetting, whenever the RBM is seeing the training samples from the new class, it learns from replays too. Roughly speaking, there are two replay types, experience replay and generative replay. With experience replay, the RBM revisits past data in its exact form. So there must be memory storage independent of the network. This approach has been widely used in machine learning, where people are less concerned about data storage, but it's not biologically plausible as it requires the storage to increase without bound. A modified way is to set up some upper limit for the memory storage. Or by generative replay, we can get rid of the storage and generate data by the RBM. Both experience replay with finite memories and generative replay allow for continual learning to some extent. The model will still forget memories with replays, but in a graceful manner, much more human-like. And we are mainly investigating these two approaches by simulations. And now I'll show you some plots. So here you can see the blue curves are for experience replays, and orange ones are for generative replays. The solid curves are the test performance after learning each new class, and the dashed curves with triangle and circle markers are two measures for replay patterns. From left to right, the number of replays is increasing. For generative replays, the RBM generates 1, 10, 100 patterns while learning each new class. For experience replay, this number is the upper limit for the memory storage. We can clearly see the replay number modulates the test performance in both generative and experience replay. The two quality measures are accuracy and diversity. The accuracy will be high if the individual replayed patterns are very similar to some original training patterns, and the diversity will be high if the replayed patterns evenly belong to all the 10 classes. As experience replay gives original samples, the accuracy is always high, while the diversity is controlled by the replay number. In contrast, generative replay always yields relatively inaccurate patterns, although the diversity is high when the replay number is low. Comparing the two types of replay, we can see experience replay is always better and can reach near perfect performance when the replay number is high. This is not surprising, as both accuracy and diversity of the experience replay is quite decent in this case. However, in generative replay, although the accuracy keeps decreasing due to the network learning more and more classes, its diversity grows high and the performance is also pretty good. This agrees with a recently developed theory that low-quality replays are good enough for maintaining old memory traces as long as each memory is replayed. By the way, here we also distinguish visible replays on the top and hidden replays on the bottom. In generative replay, this simply means the layer initiating the replay, and in experience replay, this means which layer the memory storage is attached to. Here we don't see a big difference between these two types, but depending on the parameters, distinct behaviors can be seen. I will not go to the details, but I'm happy to discuss more if you are interested. To wrap up, the take-home message is basically by mimicking memory replay in human brains, even replays of low quality, can help artificial neural networks prevent catastrophic forgetting, making the learning and the forgetting more human-like. So we think this model is also pretty good for studying memory play in human brains. That's the end of my talk. Here are some references. You can find these slides online by scanning this QR code. You can reach me via this Gmail address, or you can run all my simulations using the code here, Python code. I'd like to thank the organizers and thank you for listening. So any questions? Thank you.
Thank you, Yihu. That's the applause that you would have received from attendees if this were a real live talk, but unfortunately, this is the best we can do right now. So just wanted to let the attendees know that you can always ask question in the Q&A box, or you can come on screen. In the meantime, as people are preparing their questions, I'd like to have a much. Sure.
Should I stop screen sharing?
Sure, you can stop it.
Sure. How do I stop it? Okay, I see.
I have a general question about what is the overall goals of this type of work? So do you want to understand how memory works in general, or do you want to have a tool that we could use, say, in applications say for general applications?
Yeah, it will be more about understanding real biological replays. It will be ideal if, as a side project, we make up something and invent some good tools to be used in artificial neural networks as well as applications, because as the data grows exponentially in these days, even machines need to consider the limit for memory storage. But we as a lab, we are more concerned about replacing human brains, and we want to fit it maybe with some human data. Yeah.
Okay, so in that case, what you're showing, I mean, as long as the general principles seem to be matching how the human brain works, the performance is not as long as it kind of matches human performance, it should be fine, right? So even though the generative replay seems to perform poorer, we know from human studies that human memory is not that reliable, right? So in that case, would you say this is more up closer than when you say the experience replay?
So I would argue experience replay is something like you take a note when you are listening to a lecture. So you kind of take the exact copy of what the lecturer put on the board, on the slides, and when you before the exam, when you do your revision, it's really good to have these exact details. And comparing to just relying on your own memory. But within the brain, I don't think there's some place like a computer hard disk that you can write the exact binary patterns in that. So actually, we have to rely on generative replay in one way or the other, and that's why I think this project is really valuable, because I'm really aiming at understanding human memories.
Okay, yeah, I agree. I mean, given the interest in improving our human intelligence by some very famous people who are trying to build companies based on that, do you think this would be useful at some point in that aspect? Would you be able to improve?
You mean improve human intelligence?
Yeah, say you have an artificial implant that has this algorithm or this kind of approach.
Like some kind of brain-machine interface.
Yeah, like a memory enhancer.
Yeah, I would vote for it. I would put my bet in it. I would say, yeah, I think the kind of cyberpunk style thing would come to true before we really understanding the brains. Because I see more development and understanding in artificial intelligence, and I think machine learning can, I think machines can really be trained, and they can kind of understand our signals, but it is really hard to kind of plug in a USB disk into my brain, and suddenly I can recite all Shakespeare's books. That would be insane.
Yeah. And I just want to let the panelists know, if you have any comments or questions, please unmute yourself and ask the question. So yeah, if we don't have any more questions, thanks again for the fascinating talk here. We'll move on to our next speaker, who is Abu Faz from Alipur. Abu Faz Alipur from Indiana University Bloomington, who will be talking on the role of memory, the dorsal and ventral streams. The floor is yours, Abu Faz.
Myself. All right, can you hear me now? Okay, good. Just a second. All right. Thank you very much, Shankar, and thank you all for joining the session. And good morning, good afternoon, good evening, depending on where you are. And let's get started. So today I'm going to talk about this a computational examination of the two streams hypothesis. And these are the topics I'm going to talk about. First, I'm going to introduce the two streams hypothesis, and then I'll describe the model that we have used, and at the end, I'll present the results. So the two streams hypothesis is something that we all know. It basically says in the visual system, information is separated into two streams based on its final goal. In the dorsal stream, the information is being used for visually guided movements like motor control, things like that. And then in the ventral stream, you have information that is being used more for object recognition. And there is also this sub-hypothesis that says in the dorsal stream, the information that you have has a short memory, and in the ventral stream, the information has a longer memory. Basically, dorsal stream uses information in an online manner, and it doesn't need to retain it for a long periods of time. But in ventral stream, information is integrated across longer time scales. And if this is the case, this should provide a computational benefit. If dorsal stream really has a longer memory, therefore having dorsal stream have a shorter memory, therefore having a short memory should provide some sort of benefit for motor control, and vice versa. If having a long memory helps with object recognition, we should be able to see that. And that's the exact question we wanted to address in this study. So what we did was to build identical neural networks and train them on either visual motor tasks or object recognition related tasks, and then compare their memory spans. And specifically, the task that we have used were these: viewpoint invariant object recognition, something that you can relate to the ventral stream, or width detection, regardless of object identity, dorsal, which is something that you can relate to dorsal stream. For example, if you want to grab something, the thing that matters to you the most is most of the time is the width of the object that you want to hold on to. And the tasks that we defined were as following. We used this data set called COIL 100 dataset, which is a data set of 100 objects. And each object has 72 images from different angles with 5-degree increments. And in viewpoint invariant object recognition, what we have done was to ask the network to classify this object as what it is, for example, this granular bar, regardless of its viewpoint, like this blue row that we are showing here. But in width detection, the task of the network was to classify this width class, which is all of these objects, regardless of object identity. So these two are one class because they all have the same width. They're all 180 degrees or 0 degrees. And the networks that we have used were just super simple convolutional nets. Here we had grayscale version of whatever image that we had. These are 2 layers of convolution and then 3 layers of fully connected networks. And they're identical. One of them is being trained on object recognition. The other one is being trained on width detection. And therefore, after training, we train them first. After training, we freeze all the weights. We remove this last layer. And then we replace this last layer, if you can see here, with an echo state network or LSTM, any two types of recurrent networks. And I'll talk more about these in the next slides. All right, and now we have recurrent networks. We can, instead of one image at a time, we can feed the network sequences of images. And what happens in here is that this recurrent network could look at this history, based on the history, can tell, or with higher accuracy, what is the identity of this image or this frame. So now we have a dynamic feeding of frames to this network, and it has recurrence. The other measure that we can use is the memory dependence. For example, in echo state networks, which are basically recurrent networks with random connectivity, and nothing changes. All these connections are frozen. The input connections are frozen. The recurrent connections are frozen. The only thing that changes is the output. In here, we can manipulate how much it relies on the past memory. And basically, this is how a node in this recurrent network evolves. It takes inputs, it takes signals from other nodes, and it feeds them into this nonlinear function. Then it adds it to its previous memory, previous state. And you can manipulate this dependence on previous state using this factor that we call forget rate. And if this forget rate is 0, you don't forget anything, because this would be 1, and you multiply by your previous state. You keep everything that you had in the past. But if this forget rate is 1, this will be 0. You forget everything that you had in the past. And you only take the information from your current inputs. So the take-home message, smaller forget rates means that you will have longer memory. And we have done that. We have trained this echo state on sequences of images, either on width classification or on object classification. And then we tested the accuracy and the forget rate on the test data set. All right, let's see what happens. So we initialize the network from a initial forget rate of 0.25, and then we let it run for 18 epochs. And then in here, the blue line shows the object classification, and the orange lines show the orientation classification, or the width. And on x-axis, in here, you have network accuracy. On y-axis, you have the forget rate. So if you initialize from here, after 18 epochs, you end up being here for object classification, and for orientation classification, you end up being here. What does it mean? It means that if you want to classify objects, you need a longer memory, because if you remember, smaller forget rate. What happens if you start from 0.5 as your initial forget rate? Same thing, you drop more, and you need more memory for object classification. What happens if you start from 0.75? A similar trend. You drop to lower values of forget rate, which means you need more memory for object classification compared to orientation classification. And this is what you get. But let's see how it changes, evolves as the network learns these two tasks. I'm not going to talk about it, but if you're interested, ask the question, and I'm going to go through the evolution of this forget rate versus network accuracy through the learning, and also saying that is it a fair comparison, because network accuracies are not really equal. All right. And the next question is, does this generalize to other types of recurrent networks like LSTMs? Before being able to answer that question, we need to see how we can measure memory dependence in LSTMs. So in LSTMs, we have the forget gate and the input gate. So as a review, forget gate tells you how much of the previous time steps you want to keep. Input gate tells you how much of the current time step you want to get. And and these values are both, the forget gate and input gate, they're both between 0 and 1. If your forget gate is equal to input gate, and the ratio of these forget gate to input gate is 1, that means you're equally sampling from the past, or using the past and the present. If the ratio of forget gate to input gate is higher than 1, that means you're relying on the past more. If the opposite is smaller than 1, that means you're relying on the inputs more. And so we train the network, and we pass through the test data set through this network. And then what we do was to measure the ratio of forget gate to input gate as we pass through the test data set. And this is what we see. Let's focus on here. So on x-axis, we have the network size, and let's focus on the network size of 20 for now. And then on y-axis, we have the forget gate to input gate on a log axis. And what you see in here is that for object classification, you need a larger forget gate to input gate ratio in here. compared to width classification, which means you need longer memory if you want to classify objects compared to classifying only the width of the objects. And it's kind of intuitive because classifying objects can use the previous information as you rotate an object and feed it to the network. It can use that information, but with classification, if it wants to remember the previous time step, it's probably going to be detrimental. The only thing that it cares for is the current time steps more than often. And you see that this result generalizes across multiple network sizes, like 20 neurons, 40 neurons, 640 neurons. But the question is this a fair comparison? Are these test accuracies are similar across all networks? The answer is more or less yes, especially for larger networks of like 320 or 640 neurons. The accuracy is kind of similar. It's around more than 95% in here. And these are results of 30 different network realizations, and error bars are a standard error of the mean. All right, in summary and conclusion, two streams hypothesis assumes a shorter memory for dorsal stream. Performing dorsal stream functions seem to require a shorter memory span, and this effect holds across echo states and LSTMs. And thank you for listening, and I'll also want to thank my supervisors, and you can connect me through GitHub, Twitter, email me, and the paper with more details of this study is posted on bioRxiv, and you can scan this QR code for that. And thank you.
Thanks for the interesting talk, Abul Faisal. And I'm just reminding the attendees that you can always ask questions using the Q&A panel, or let us know if you want to come on screen. Yeah, as we are waiting for questions, so you briefly touched upon this, that you need more, or having a longer memory helps with object recognition versus I didn't quite get what the second for the
orientation or width.
Yeah.
Orientation.
Orientation, right?
We use them interchangeably, but yeah, or we can say orientation.
So would you say like the feature size for each of the actual information, the feature size of the actual information would also be different?
What do you mean exactly by feature?
So like for an object, let's say I need to know, let's say the shape, the size, the colors, whereas for orientation, I just need to know which way it is directed. I don't really.
Right. That's right. So there are fewer number of features that you need to have in order to perform orientation classification compared to object classification. That's right. Okay, so that's possibly the main reason why you need longer memories, or potentially. And the other intuitive explanation would be that, well, for example, if you give these sequence of images to a network, this is the first image is this, the second image is this, the third image is this one, and the features are changing constantly. And if you want to keep memory of the previous images, it's going to interfere with your ability to classify the width, because the only thing that you care about is that, how much is this? Is it like 180 degrees, or is it like 45 degree, or so on and so forth. But in here, when you want to recognize objects, for example, this one can clearly show you that this was a granular bar. You want to hold on to this information because it's going to help you in the next and upcoming frames. And that's going to help you if you have a memory of the previous frames. But in here, having a memory of previous frames is not that informative.
Got it.
And it kind of goes with the intuition of dorsal ventral memory spans. If you want to go and grab something with your hands, you don't really need to have the object identity information. You just need to get the width information to adjust your grip aperture. But if you want to recognize objects, you need to have a longer memory. Yeah.
Okay. And so I think I missed some of the early parts, like having some trouble with the Zoom here. So you extract features from this to feed to the network, or do you send in the entire image to the network?
I send the entire image to the network. So this is like a front end of the network, and this is the image. This is basically image grayscaled and downscaled to 28 by 28 pixels, and then it's fed into a convolution layer, and then max pooling, convolution, max pooling again, fully connected, so on and so forth. So it's basically fed the image grayscaled.
Are these two networks are identical except for the final layer? Do they share weights?
No, they don't share weights. They're just randomly initialized, but the architecture is identical, as you said. The architecture is identical, but the weights are randomized, and we have average across 30 different network realizations for calculating the results. Yeah.
Is it possible to do weight sharing? Because, yeah, I mean, so how can a brain tell which pathway should I go without pre-processing the entire?
Mm-hmm. So that's a good question, and in here the assumption is that well, more or less it's more of an anatomical. The assumption in here is a loose assumption, and that's right. But the assumption is here through development, these are separating into two different pathways.
Okay.
Yeah. It's more of a developmental genetic thing that it's already through evolution. It has evolved to be really good in doing this task versus the other task.
Mm-hmm.
Okay.
Yeah.
All right. So if we don't have any more questions, we can move to the final speaker of today's session. We have Thomas Folders also from Cardiff University, who'll be talking about modeling sleep-dependent memory consolidation. Take it away, Thomas.
Thank you. Let's see this one. Hopefully the computer. Yeah, here we go. Greetings, everyone. I'd like to speak a little bit about my foray into connectionist modeling in an attempt to better understand the tasks that our lab is working on, to complement the behavioral work that the lab has been working on, and basically my experiences with learning about how computation and representation are interrelated in neural networks. Started out my work focusing on the complementary learning system, and most of you will be familiar with this. It's a very strong computational framework that focuses on the separate roles that the hippocampus and the neocortex are doing in trading off over focusing on pattern-separated representation in the hippocampus and more overlapping code in the neocortex. And the idea is that the brain is best at solving these two separate memory tasks, basically the generalization part and the extraction part, and the avoiding on the interference part, by using two separate systems for this, a fast learning one and a slow learning one. Classically, the idea is that in the complementary learning system, it is during offline consolidation that the hippocampus basically slowly teaches the neocortex and does the extraction during that time. But more recently, with the advent of a lot of sleep-dependent memory consolidation research, some formal theories are going further, such as the IOTA model, for example, and try to hypothesize about the separate roles of the given sleep stages and what these might be doing. And one such computational outline of a theory argues that sleep-dependent memory consolidation, so basically the performance increase that we see after sleep for certain memory tasks, could be well modeled in some sort of sparse autoencoder. However, the picture is fairly complicated. In recent reviews, for example, in one review by Lerner and Gluck, that looked at more than 40 sleep-dependent memory studies, focusing on people's performances in extracting hidden regularities. The actual role, the actual task-dependent effect and sleep stage-dependent effect is still fairly murky at the moment. But some things that looking at the literature are visible at the moment is that there's a very strong task-dependent effect. So it's more likely that a given task would be replicated as far as its sleep-dependent effect is considered than its category of task. So the paradigm seems to matter a lot. As far as explicit rule extraction performance is considered, there does seem to be an added benefit of having a temporally a task that involves some temporal regularity versus one that is just stationary. And what's also interesting is that they argue that a lot of sleep-dependent memory consolidation effect could be explained by local memory strengthening mechanisms, which is actually quite convenient from a connectionist modeling perspective, as we will see. As a first attempt in outlining a connectionist sleep-dependent memory consolidation model, we were focusing on associative inference tasks. And ideally, we would use as a start, as a seed of the model, we would use some sort of formal framework that can account for different kinds of behavioral paradigms, such as the transitive inference task, paired association, acquired equivalence. And one such theory that we were attracted to in the beginning was a model called remerge, which is a very simple, basically two-layer neural network that involves some sort of competitive inhibition in its hidden layer and has a recurrent bidirectional dynamics between its two layers. I will be focusing on the transitive inference task in my work so far. And basically what we need to understand is that in the transitive inference task, people are shown pairs of items, A, B, C, C, D, E, F, and so forth. And the assumption is that through some sort of Hebbian learning mechanism, they're able to form conjunctive representations, supposedly in the hippocampus. And by just making this very assumption, if we assume that there's competitive recurrent dynamics involved in the network is able to do inferential behavior, exhibit inferential behavior. So if we look at, for example, a BD trial, we would, one second, we would stimulate our network's feature layers, and indeed we would see that it would settle in a local attractor state where both B, C, C, D, and the D node are all active. And then the response layer would basically be responsible for choosing the leftmost or the rightmost relational item in the linear hierarchy. So this is one conceptualization of how generalization might work in this task. Something it's called the retrieval-based generalization. But how does this relate to sleep? In the paper, Kumaran and McClelland argue that basically we could account for the sleep-dependent effect in the transitive inference task, which we can see here from a classical 2006 study, how that might look like. People, before they go to sleep, perform very good on the premise pair part, but are not yet good on the inferential task of the performance, so they're not able to make transitive judgments that involve items that are 1 or 2 degrees separated from each other. But after 12 hours of sleep, we'd see a significant inferential boost. So this is the sleep-dependent memory effect of transitive inference task. And they show that the network is able to account for this effect just by strengthening its premise pairs. So just by local Hebbian strengthening, we're able to account for this effect. Now they don't show, the remerge model is a model of retrieval. It's not a model of learning. So first we wanted to find a network or a learning algorithm that would be able to learn these very sparse connections that are necessary for the remerge to work. And we tried different algorithms. A lot of them didn't work. And we focused on contrastive Hebbian learning, which here in the plot we see called referred to as GeneRec. But what wasn't clear for us in the beginning is that competitive inhibition is crucial for robust generative performance to emerge from the network. Later on, finding a 2001 article that shows this effect quite nicely. A combined learning algorithm, a learning algorithm that combines Hebbian learning and contrastive Hebbian learning, called the ABRA, was actually necessary in our findings to have a network that is able to replicate those sparse connection weights that the remerge needs to function. And the idea, the reasoning behind why this competitive inhibition is important, and there's many different kinds of inhibition algorithms that one can use when trying to model a cognitive task, cognitive experiment. The K-winner takes all inhibition that is involved in the Libra algorithm seems to be at a very good crossroads in that it's able to take advantage of contrastive Hebbian learning, so it's able to do error-driven learning in a non-supervised way. But thanks to a Hebbian component in the learning algorithm, which you can see here, and competitive inhibition as biasing factors, it seems that it's much more able to basically settle into local attractor states and separate local attractor states much better than contrastive Hebbian learning alone, especially in tasks that need some sort of componential learning, where tasks where separate components need to be separated from each other as soon as possible. So in order to take advantage of this algorithm, we try to model the transitive inference task in a cortical hippocampal model of the similar sort. We used the classical O'Reilly hippocampal model, but we lesioned the trisynaptic pathway, leaving only the monosynaptic pathway, meaning the entorhinal cortex and the CA1. And basically, these are the results that we looked at as a first attempt. So one interesting thing is that we found that if we compare the performance of the model on the transitive inference task, if we look at it with constant cortical inhibition versus something that's akin to a sleep-dependent inhibition, we see that the sleep-dependent inhibition seems to improve. So it does make an added difference if we change the inhibition during halfway the learning trajectory. That's one interesting thing. The other thing is that we were able to replicate, with this manipulation, we were able to replicate this, the Ellenbogen effect on transitive inference and sleep. And more interestingly, we've also found that the Libra algorithm is agnostic to whether the generalization happens during encoding or during retrieval. And that is very much in line with the still open debate about whether the relational theory or a more retrieval-based mechanism explain sleep-dependent generalization or not.
We are almost in the end.
Yeah, sorry. This is my last slide, and these are my citations. Thank you.
Thanks for the wonderful talk, Thomas. Sorry I had to interrupt you. I mean, we're
No,
almost at the end of our session,
was...
and we need to, we might get kicked out of the room when the next session starts.
Right.
So as we are waiting for questions, so I was wondering, like, I didn't let you finish the last section, which is very interesting, that it's able to account for both the two types, like whether if it is happening in the retrieval or in the encoding. So how do you, like, do we have any empirical results suggesting that like one is more prevalent than the other, or is it's still unclear?
To the best of my knowledge, there seems to be a lot of empirical results where people try to tie their findings in the discussions of their papers to one or the other paradigm. I feel that we just don't have enough resolution to really distinguish between these two paradigms yet. But it's definitely reassuring that that as far as the learning algorithm is considered, it doesn't seem to matter from a neural network standpoint. Yeah, we're going to need some extra constraint.
Okay, got it.
Yeah.
Yeah. And yeah, so if other panelists have any questions, just reminding, you can always unmute yourself and ask questions. And attendees, if you want to come online, just let us know. So the reason I'm kind of curious about this is I did memory, like I work with humans and I do stimulation experiments in humans. And typically what people say, like when they study memories, they try to look at only at the kind of like the retrieval phase, or like the phase where they have to keep it memory. But this is a working memory, of course. It's not like a episodic memory. The period where there is a clear encoding period where they are seeing the stimulus, and then there is a short time period when they don't see the stimulus, so they have to kind of keep it replaying. So in that case, the way this setup is formulated is the learning happens in that interval when they are not seeing the stimulus, right? Or would it be even when they're seeing the stimulus?
Can you say again? So it's a
when
classical neural network type setup, right?
Oh, sorry. This is a complete experimental in humans. They're doing a memory task, but this is a working memory task.
Okay.
I'm just trying to understand, like, when you say encoding, I'm trying to understand how.
Oh yeah, I see. Yeah, I know, because it's different for a working memory person. I think it's much more nuanced. I guess in that field. Yeah. So here encoding only means in neural network sense, it only means whether if we control the competitive inhibition in the neural network during the training phase, or if we keep that static and only alter it at recall, right? So is it part of the weight updating process, or is it only part of the recall? So we're only manipulating the recall environment, and then the weights are obviously frozen in that sense.
Okay.
So that's how.
Okay.
Yeah, that's the way we can distinguish between those two paradigms here, as far as the neural network sense is considered.
Yeah. Yeah, that's really fascinating. But I think we are almost out of time. So before we get kicked out, I just want to thank the panelists again, and thanks all the attendees, and hope you have a good rest of the day, however long that is. All right.
Thank you.
Bye-bye.
Thank you.
Thank you.
Goodbye.
Okay. So it just takes a few moments for the Zoom webinar to let everyone through. So I'll give it one more minute and then I'll introduce the session. All right, so I'm going to go ahead and get started introducing the session. So hello, everyone. I'm Adam Steele. I'm a postdoc at Dartmouth College in Hanover, New Hampshire, USA. While people are arriving in, I'm going to go ahead and just say, use that opportunity to tell everyone in the United States to make a plan to vote. If you've got a plan to vote, please call a friend and make sure they have a plan to vote. And if you're not in the US or you can't vote in the US elections, please call one of your US colleagues and make sure that they have a plan to vote, because it's super important. So I think we've got a few this is a great session coming up about visual navigation and visual motor behavior. I think it's going to be an awesome session. Just to remind everybody, I think we're all used to it by now. But if you have questions for our speakers, please use the Q&A. That's going to be open at any time. Also, you can upvote your favorite questions to make sure that they get asked live. You can also use the chat. We'll be monitoring the chat the whole time. So if you have any questions or you want clarification, you can go ahead and post in there. Also, if you would like to right now, post in the chat just to introduce yourself so that way we all know who's in the room. That would be awesome. So just, write your name, maybe where you're calling in from, so that way everyone knows who's all watching. So with that said, I'm going to get started with the session. Our first talk is from Lillian Chang from the UC Irvine. And her talk is titled Travel Direction as a Fundamental Component of Human Navigation. Oh. So Lillian, the floor is yours.
Okay, thank you. Can you all see my screen now? All right.
Yes, we can.
Started. Hello, everyone. I'm Lily Chan from the University of California, Irvine. And today I'd like to talk about my study, travel direction as a fundamental component of human navigation. Imagine you're on your way walking to a Starbucks and you can hear birds singing up in the tree and feel the breeze touching your skin. And suddenly you notice a bunny running by. Where did it go? You turn your head to follow the bunny running and hiding into a nearby bush, while your feet are still walking toward your destination. In this case, where you are facing toward and where you are moving toward are both part of your sense of direction, but they represent different spatial information. Head direction refers to where someone is facing toward, while travel direction refers to the direction of one's body movement. In daily life, people usually conflate the two directions, thinking that where we are facing toward is where we're going. But as you have seen in the bunny example, these two factors can offer different spatial information. Extensive studies have focused on head direction cells in the brain. Although head direction has been found to be a crucial part of human navigation, travel direction might be more important to determining the travel trajectory. However, it is unclear whether travel direction plays a primary role in navigation. As travel direction forms a travel trajectory together with speed and time, we hypothesize that travel direction is a fundamental component of the internal representation system of human navigation. To test this, we adopted a visual motion adaptation paradigm, much like the waterfall illusion, which you can try yourself by staring at it. In motion adaptation, neurons selected for visual motion features are theorized to adapt to visual stimuli that corresponds to its selective firing properties after prolonged exposure. This adaptation often results in a decrease in the neuron responsiveness to the same stimuli compared to an unadapted response. This is represented at a behavioral level as a motion after effect, such that a stationary stimulus will appear to move in the opposite direction to previously viewed motion, just as you perceive the waterfall going up once the video paused. Therefore, we used a motion adaptation paradigm to probe for selectivity of travel direction. In our experiment, we predicted that navigators adapting to a travel direction will show an increase in reporting the opposite direction. We carried out our study in a virtual environment, which includes a virtual hallway and 2 reference points indicating cardinal directions, a sun and a moon, that located at the 2 ends of the hallway and were said to be infinitely far. Our experiment had 2 sessions. Each session was conducted on a separate day. One session was an adaptation session. The other session was a control session. The order of the 2 sessions was counterbalanced among subjects. For the adaptation session, half of the subjects were randomly assigned to adapt to the moon direction, and another half were assigned to adapt to the sun direction. Control conditions were the same across all subjects. Now, let's look at the adaptation session. In the adaptation session, we first adapted people to visual self-motion in one direction in a virtual hallway, either towards a sun or a moon. The head direction was reversed occasionally to be dissociated from the travel direction, while travel direction during the adaptation phase remained constant in one direction. Changing head direction precluded optic flow from contributing to any observed adaptation effects. We then tested people with a series of visual back and forth movements toward and away from the initial travel direction. The head direction also reversed occasionally during test as well. At the end of each test trial, we asked subjects to report their net travel direction. We varied the percent of movement toward the sun direction during the test phase between 20% and 80%, allowing us to measure psychometric functions for perceived travel direction. After each test trial, we presented a 10-second long top-up adaptation to maintain the adaptation during testing. And after each top-up was another test phase trial, followed by another top-up adaptation, and then a test trial, so on and so forth. Now, let's look at the control session. In the control session, the viewer was stationary during the adaptation phase. To match the adaptation session, head direction in the control session also occasionally reversed. The test phase of the control session was the same as in the adaptation session. We compared the adaptation session with the no adaptation control. When using the percentage of the actual and reported movement to the adaptation direction as X and Y axis, we predict a line for the control session where the reported rate increases with the actual rate. For the adaptation session, we predict the adaptation line to be a line that is shifted to a lower position than the control line because people tend to have weaker response to the adapted stimuli. Here are the results for the control session, shown in red, and the adaptation session, shown in blue. We found a difference between the people's report of the net change of travel direction in the adaptation session and in the control session, as you can see in the difference between the two lines. The results indicate a motion after effect of travel direction, suggesting that travel direction is a fundamental component of human navigation. Interestingly, subjects had a higher frequency of reporting net travel toward the adapted direction, indicating that the after effect is opposite to the traditional motion after effect. We have run additional controls and conducted additional analysis, indicating that this is not driven by response bias or other factors. And I'm happy to answer questions about this in the discussion. Overall, this study suggests that travel direction is a fundamental component of human navigation. We will next investigate whether these signals can be detected in the brain during fMRI imaging. If you'd like to know more details of the study, please scan the QR code for a poster version of the study. Thank you for your listening, and I'd like to take questions.
All right, thank you, Lily. That was a really great talk, super clear, and I especially appreciated your lead in with the hypothesized results. I thought that really was helpful for me to understand. So I have a few questions, but if anyone in the audience would also like to ask a question, please feel free to post in the chat or in the Q&A. So one of my questions was more of just a comment about how interesting it is that you point out that travel direction and head direction are often decorrelated. I think it's especially important in the study of human navigation when we use virtual navigation. And I'm reminded of like how when we play video games, for example, and also navigate on like a desktop computer, that is a specific time when often the facing direction can be decorrelated from the head direction. So like like many times you would be strafing as a character more often in a video game than you would in real life. And so I was wondering whether you guys have considered how frequent, like any kind of naturalistic behavioral analysis or in either humans or animals to look at how often in like real world navigation, heading direction really is decorrelated from facing direction. Do you have a sense of that?
Yeah, I think part of the reason for us to do it in VR is just because it's easier to control it. And yeah, in real life, people do look around, but not like looking around, like going like a crab way, only looking in the same direction and going in the other direction. It's hard, it's not very natural for people, but I think maybe with more immersive VR headset, it could be possible to conduct, or maybe like a well-round treadmill that people can just
actually it was sorry, my headphones have dropped off for a second.
I actually I'd been speaking recently with somebody who works with an omnidirectional treadmill, and they said that one of the nice things is that people behave more similarly to real world navigation, where they realign themselves before they make a movement, unlike in like a video game task where they can easily like shuffle. And like a lot of times I think about the times when people do turn their heads and navigate when they aren't looking in one direction. And it's similar. I think your example of the rabbit is a good one, where you're like tracking something. And that seems to generally be the case. It's like I can think of two examples off the top of my head, which is the example like you did with the rabbit, or like generally tracking something, or in sporting events when you might be tracking something too. So it seems like that's part of the behavior. My other question was leads in nicely to your future direction, which was whether you think that this is going to be a separate neural system that underlies or underpins this travel direction maintenance from the heading direction system, or the and what systems you might expect that to be? Like where do you think this is going to be coded?
Yeah, so by saying heading direction, it's actually already conflating the head direction and travel direction. So
right.
So let's separate them. So
sorry. Yeah, well, my question was poorly phrased.
Yeah, for the head direction, I know what you're saying. So yeah, so I think because people have conflated it before, so it is very likely that the head direction system that we already have found, that like subiculum, thalamus, retrospinal cortex, these regions could have some overlap with the travel direction system, or travel direction system is a subset of it, like a special case of it. Because I think travel usually has to involve motion, so I think the high-level visual motion system could also play an important role here. So there should be some interaction where different parts of visual system and like directional system involved in it. That's what I'm thinking, but I'll see what I can find.
Cool. Well, I'm really excited to see the next phase of the results. So thank you very much for that talk. So with that, I think we'll move on to our second talk of the session. This is going to be given by Michael West from Massachusetts Institute of Technology, and his talk is titled Role of Path Information in Visual Perception of Joint Stiffness.
Can you guys see my screen?
Yep, you're good. Yep.
Cool, okay. So thank you for the introduction. I'm Michael West. I'm a third-year graduate student at MIT. This project was in collaboration with Professor Megan Huber at University of Massachusetts and my PI, Neville Hogan at MIT. So just to motivate the idea of this project, humans have, if I can play this video, humans have a very good ability to extract latent information from visual observation of biological motion. This is best shown in point light animations, where you can see in this video someone who is trying to jump. Previously, the person was walking, and climbing stairs as well. And this idea has been advanced such that people can determine age, gender, even emotions from these point light animation videos. However, we came to ask, can humans perceive limb stiffness from visual observation of motion? And we believe this question is important for two reasons. One, we have found that modulation of stiffness, or more generally mechanical impedance, is important for neuromotor control during physical interaction. And two, identifying abnormalities in muscle impedance is often used during rehabilitation after someone has lost motor function. Thus, we hope extending this work can provide a new rehabilitation method. So previously, we ran a set of experiments where we simulated an arm motion that had varying stiffness. We let subjects view multiple simulations, and we found that yes, subjects can estimate changes in stiffness from visual observation of motion. You can check our previously cited work here. However, the question still remains how. And we hypothesize that this is due to the relationship between motor action and motor perception. Furthermore, we believe that our model used to simulate this motion was congruent with how humans produce their own motion, which I'll go into greater detail later. Nonetheless, we're still unsure what motion cues humans are using to estimate stiffness. So in a set of new experiments, we aim to answer that question. Specifically, we draw upon the literature of the 1/3 power law, which is basically a mathematical relationship of the velocity profile during upper limb reaching. And we hypothesized that in our previous experiments, our simulations typically did follow this power law, so we've hypothesized based off of this literature that manipulations of the velocity profile that violate this 1/3 power law will inhibit subjects' ability to estimate stiffness. So as mentioned before, we found that subjects can estimate stiffness using only visual observation of motion. And this is a remarkable feat when you consider the definition of stiffness, and that it's the relationship between force and motion. And in this experiment, subjects had no access to force information. So in a series of 3 experiments, where we either modulated both the shoulder and elbow stiffness, or just the elbow stiffness or just the shoulder stiffness, we found that subjects were able to increase their stiffness ratings with the simulated joint stiffness. So there's this positive linear effect of simulated joint stiffness on rating. And this is our previous work, but the question still remains how. And I think to answer that question, we need to draw upon the underlying controller that was used to simulate these joint motions. So here we see this complex control law, but really it's quite simple. What it does is it has the superposition of two impedances that act as separate attractors. And then the first one is a spring damper system that pulls the endpoint of the arm around a nominal trajectory, which in our simulations we define to be a circle. And that's what these two terms take care of here. The second attractor is the stiffness at the shoulder and elbow joints. And what that does is pull the arms to a nominal configuration. And this is the stiffness in these joints is what we change. And that's taken care of in this term here. Specifically what we modulated with this was this KQ value, And that is what we found subjects were able to, that's what we hoped subjects would be able to tease out from our simulations. And that's what we found as well. So the fact that humans are able to estimate changes in joint stiffness from visual observation and motion leads to two key conclusions. One, humans assume stiffness affects motion. And two, it is likely that humans produce their own motion. It is likely that how humans produce their own motion is congruent with the control law used in our simulation. This comes back to a relationship between motor action and motor perception. We believe that if this controller can be perceived, it is likely that this controller tells us something about motor action as well. If subjects are using, and that's under the assumption that subjects are using their knowledge of how their own control, their knowledge of how they produce their own control when they're perceiving these simulations. So just to summarize, this control law is very complex, yet subjects were able to identify a single parameter or identify changes to a single parameter within it. And again, the question still remains how. And what we aimed to ask was, are they looking at past information or is temporal information important as well? And the way we did this was through two methods. And the first was, while keeping the past information the same from our previous experiments, we manipulated the velocity profile, the temporal information. And what that means is, generally, and in our first simulations, it's been consistent with the literature of this 2/3 power law, which is just this mathematical relationship here. And what it relates is radius of curvature to velocity via this exponent beta, which usually equals 1/3. And in our biological case, what that simply means is that when you're moving around an ellipse path with curvature, you go faster around the straight edges and you slow down around the curves. So what we did was, in these new simulations, we manipulated that velocity profile such that in experiment 1, we had a constant velocity where we set beta equal to 0. In experiment 2, we set beta equal to -1/3, where there's this reverse velocity radius of curvature relationship. And what that means is subjects are going slower around the straight edges and faster around the curves. And then experiment 3, there was no velocity radius of curvature relationship. And I'll explain how we manipulated that a bit later. The second thing we did was we asked subjects to write down the strategy they used to estimate stiffness. And we were trying to see if subjects could articulate what it was and if we can gather information from that as well. So our new simulations took the same path, but recall the video that I showed you earlier, subjects were they could not see this path. They could only see the motion of the arm. And we took the experiment where we kept the shoulder stiffness the same and modulated the elbow stiffness. So we had 30 new subjects, 10 for each of the 3 new experiments, and they watched 30 arm simulations, each with, and the arm simulations had 6 different elbow stiffness conditions shown here. And they saw each condition 5 times in the block randomized order, and then they rated their stiffness on a scale from 1 to 7. So you can see here in our new experiment 1 that the velocity is constant all around the path, right? In experiment 2, we see that subjects are going slow around the straight edges and faster around the curves. That's a reverse velocity curvature relationship. In experiment 3, there's no parallel relationship here. What we did was we took the time series that was computed to make a constant velocity, tangential velocity around the curvature for the 50 Newton meter per radian stiffness simulation and applied that to all of the simulations as well. So there was no velocity curvature relationship in experiment 3. And what we found was actually really interesting, right? So we hypothesized that manipulations of the velocity curvature relationship would affect subjects' ability to estimate stiffness, but what we found was the opposite. It did not. So subjects were still able to to increase their stiffness ratings based off of the simulated joint stiffness, despite these velocity manipulations. What we see here is all the subjects' individual data, and you can see the simulated joint stiffness and their chosen stiffness rating. And what we did was we fit a linear model to that, and we defined the goodness fit of the linear model by the coefficient of determination, and we determined that we use that as a metric of subjects' performance. So a perfect linear relationship would mean, would be a high coefficient of determination, means subject did the task very well. And using the coefficient of determination R squared as a measure of subject's ability to estimate stiffness, when we go ahead and look at the distribution of this R squared value across the 3 new experiments and the original experiment, what we found was experiment had no effect on subjects' ability to estimate stiffness. So subjects were able to estimate stiffness equally the same regardless of how we are statistically equally the same regardless of what manipulations we made to the velocity profile. And remember, the second thing we did was after subjects completed the study and saw the simulations, we asked them to write down the strategy they used. This is an open-ended question. Subjects could have said anything. Some subjects even drew pictures. What we wanted to do was tease out the information. We were specifically looking at whether subjects used path information, temporal information, joint motion, or endpoint motion. And we codified that just that there was a yes or no for each one of those quantities. And what we looked for path information specifically was the words distance, displacement, range of motion, angle. For temporal information, speed, rate, acceleration. And for joint motion, joint, shoulder, elbow, angle. And then for endpoint motion, endpoint or hand. And then also if subjects drew a picture and pointed to the endpoint or the joint, we were able to codify that as well. And what we found was that most subjects reported using path information and joint information. However, many subjects could not actually articulate what strategy they used, so their response was not codified in any of these ways. And the most surprising result is that none of the 4 motion features that subjects used actually were significant predictors of subjects' ability to rate stiffness. So if you look at the motion features that subjects reported using and their R squared value, or the coefficient of determination, which we decided is a measure of how well subjects are able to do the task, we can see this wide range of performance. So this kind of tells us that, okay, subjects may not necessarily be able to articulate what it is that they're doing. We have hypothesized that this may be because estimating stiffness is something that's so fundamental to the internal model. It's something that we can't necessarily articulate what we're doing because we're doing it very innately. So to conclude, we found that humans can correctly infer changes in limb stiffness from non-trivial changes in multi-joint limb motion, and they can do this despite the manipulations to the velocity profile. And this suggests that stiffness, or more generally mechanical impedance, is encoded in subject's internal model used to do this task. And secondly, it suggests that path information predominates temporal information in the visual perception of stiffness. And what exactly did we do here? We used perception to learn more about motor action, right? So we use this idea of, can subjects perceive this controller? Well, if they're using what they know about their own motion, and if they can perceive this controller, it's likely that this controller is somewhat congruent, or there has some congruencies with the motor action. And then secondly, we believe that by answering the question, how are subjects estimating stiffness from visual observation, we can kind of move towards a new, less encumbering method of rehabilitation for those who have lost motor function. Just want to acknowledge my collaborators, Professor Megan Huber at UMass Amherst, and my PI, Neville Hogan, on my funding sources. If you have any questions, feel free to contact me. My email is amwestjr@mit.edu. Thank you.
All right, thank you, Michael. That was great. That was an excellent talk. If anybody has questions, you can go ahead and post them in the Q&A. I'm going to go ahead and get us started with one observation that I had, which was that path really seemed, so we didn't have an example, a visual example of what the changes in speed or temporal information, as you called it, would have looked like, but it really seemed like the paths varied dramatically across stiffness conditions. So that seemed like a very salient manipulation. I'm wondering if you had a way of making the temporal information, of altering the, if you altered the temporal information in some way, could you change the stiffness rating? Like if you made, instead of smooth transitions in speed, if you had made the transitions in speed like more jerky, or you added some stochasticity to the movement, whether that would also be interpreted as stiffness. Does that make sense?
kind of. I guess your question was two parts. So I guess
Yeah.
the first part, you said you didn't see a good explanation of what exactly was going on. Here's a visual of all the simulation that subjects would have seen just by each experiment. But in the second part, you kind of asked, well, can you repeat the second part again?
So I guess I'll rephrase the second part as, do you think that there's a temporal manipulation that you could apply that would also look like stiffness? So if a person who's has extreme joint stiffness might be able to make a full circle, but it could be like a little bit more jerky in the movement, like maybe there's more stopping and starting. So it's not in the way that you had two very smooth like acceleration and decelerations. If you added more stochasticity to the speed of the arm movement or the limb movement in this case, would that mimic or make somebody have a similar effect and make somebody think that it's a stiff joint?
Yeah, so I guess what you're saying is, can you find a velocity profile such that subjects are unable to actually estimate stiffness? And I think,
or is there a different? It's not necessarily can you trick them into not being able to estimate stiffness, but more that is there a different velocity profile that we would associate with stiffness? Like, does velocity have a specific profile that we think of as stiffness? Does that make sense?
Yeah, I would say that and to that specific question, these results would suggest that no, right? So we've been able to manipulate the velocity profile to something that humans don't typically do. And we've done it in 3 different cases, and we found that consistently subjects are still able to estimate stiffness. So this tells us that subjects probably aren't using velocity information to estimate stiffness, right? They're using motion information. And I think it's possible that, we could find, it's not something that's falsifiable, saying that, there's not a such,
yeah.
I can't definitively say there's no velocity profile that doesn't work.
Yeah. So one question just popped up in the Q&A. So you can address that during the next talk, but we're going to have to move on, unfortunately. But yeah, so if you can go ahead and type your answer to that question there. But thank you again. That was really very interesting work there.
Thank you.
So we've got a final talk in this session from Aya Alwan from the Ohio State University. And her talk is titled Stability and Control of Walking at Various Speed Slope Combinations.
Thank you, Adam. My name is Aya Alwan, and I'm a PhD student at the Mechanical Engineering Department at Ohio State. And I'll be presenting my work regarding investigating stability and control of inclined walking for healthy subjects. So walking, sorry, let's see. I think it jumped. All right, so the motivation for my work is that 1 in 4 older adults report falling each year. So falls are a leading cause of injury-related deaths, especially for older adults that are 65 or older. So by understanding the mechanics, energetics, and cognitive behavior of human movement, we can hopefully predict fall better and consequently lower the risk of falls by designing better assistive devices, prosthetics, and exoskeletons. Our work also impacts fields such as robotics and rehabilitation. So why study sloped walking specifically? As you may know, slopes are often used as an alternative to stairs for special populations such as the elderly, lower amputees, and people with lower limb deficiencies. And such tasks can be challenging, especially that muscles need to generate more power to the legs when walking uphill. So hundreds of papers exist on level ground walking, but unfortunately few of them focus on the control and stability of walking on slopes or uneven terrain. So walking is not perfectly periodic, even on a treadmill. Small perturbations usually generate from muscle and sensory noise as part of natural variability. Those sort of perturbations actually increase with aging, and we know that from previous research. So how do humans recover from these deviations? There are a couple of ways. One is foot placement modulation in the direction of the perturbation. So for the second video here, you see that the experimenter is changing belt speed to basically simulate slips or trips. And to recover from such perturbation, the subject is placing their foot in the direction of that deviation. In the first video on the top, you see subject running, and this is another way of recovering from deviations, which is like applying more leg force or push-off impulse modulation. So here we will investigate control strategies of healthy adults. We will investigate the control strategies that healthy adults use when walking on uphill slopes to stabilize their gait. So the first part here, I will present to you the experimental analysis. Our lab uses equipment such as motion capture system, an inclined treadmill, and reflective markers. We collected data for 10 subjects, 4 females and 6 males, for 2 minutes each trial. We have collected 12 randomized trials for 3 speeds, 0.7 meters per second, 1 meters per second, 1.3 meters per second, as well as 4 different inclinations, 0%, 3%, 6%, and 9%. For the purposes of this presentation, I will only be presenting results from 1.0 meters per second and results from the left-to-right transitions, which we know that are mirror symmetrical to right-to-left transitions. To infer control, we use linear regression methods. We define foot placement gains as output over input, or in other words, the coefficients of the following linear models. So in the first function here, or the first linear regression here, you see that the output is the sideways foot placement, and the input are the hip states in the sideways, fore aft, and vertical directions, the positions, and as well as the velocities. In the second equation, you see the fore aft foot placement equation, which we use the fore aft foot position as the output, and again, the hip states are as input. So in the video here, you see that we're pulling the subject sideways from either side, and the subject is recovering from such perturbation by placing their foot in the direction of the pulling or the perturbation. So what does that mean for us? That tells us that deviations of foot placement from the average values can be explained by deviations in the pelvis position and speed from average values at mid-stance. So from our results here, I'm showing on the left side the response of sideways foot position responds to sideways hip position for the lowest inclination, the 0%. On the right side, you see the same information for the 9% inclination. Within each plot here, you do see a spread, and the p-value is significant. And that we know from previous studies is that the hip foot position is explained by deviations in hip positions. However, when we did t-test between both of these values or both of these plots, we didn't see any significance. And similarly for the fore aft foot placement, we did not see any significant from the lowest slope and the highest slope. Sideways hip states slightly increase with steeper inclination, but not significantly. So in this plot here, on the x-axis, I show the number of steps. On the y-axis is the sideways hip position deviations, delta x hip. And each column here, you see a different inclination that the subjects were asked to walk on. So let's compare the 0% to the 9%. You do see a little bit of a spread, but it's not that significant. Similarly, for sideways hip velocity deviations on the y-axis here, you do see more spread as we increase inclination, but it is not significant. We also see a similar trend for fore aft hip velocity. We also quantify stability by calculating flow-K multipliers. Flow-K multipliers are 3 state variables that tell us how quickly small deviations are corrected. So if the number is less than 1, then the system is stable. If the number is greater than 1, we know that the system is unstable. The way we calculate it is by calculating individual step-to-step mapping from each transition. And multiplying those individual mappings will give us a full point Cray map. Taking the eigenvalues of the full point Cray map will give us flow-K multipliers. So here you see the equation that's showing inputs and outputs linear regression and the Jacobians as a function of that. So flow-K multipliers for our specific experiment show a little bit of trend towards instability, especially for flow-K multiplier 3. We see a little bit of trend, however, not very significant. And the overall number is still less than 1. So you see the highest is about 0.35. So the system is still overall stable. The second part of my study is building a data-derived feedback controller, and this work is in progress. We simulate the human body as an inverted pendulum biped model. So the body is represented by a center of mass with massless legs, and in between the push-off and the heel strike phases, we represented by an inverted pendulum. So using experimentally derived linear models of coefficients, we can modulate the magnitude of the push-off and the step length to keep the biped stable. Such methods will help us simulate sloped walking for different inclination values and detect when the control becomes unstable. And then we can modulate the input to make the controller become stable again. So my hypothesis is that for much steeper inclinations, we may find that the controller becomes highly unstable. Future work can include conducting walking studies with healthy adults in comparison with older adults for different types of everyday tasks, including slopes, uneven terrain, unexpected obstacles, external forces, and so on. We can also build a comprehensive predictive model of walking that can encompass different robust adaptive control stability properties of young adults versus elderly by considering multi-body dynamics, different body segments, and not just simplified. We can also consider energy expenditure and muscle forces that result in certain gain behavior, especially that we know that humans walk in a manner to minimize effort. I'd like to give thanks to Manoj Srinivasan, who is my advisor, and Nidhi Sitapathy, who is a current postdoc at University of Pennsylvania. And I'm open to all questions. Thank you.
All right, thank you so much. That was a wonderful talk. I really liked your simulations at the end, and your future directions seem like a good, like they're really building on nicely from the data that you've recorded. So if anyone has Q&A or questions, rather, you can post them in the Q&A. I wanted to just go back to your future direction. We do have one question that's come in. How do you expect your results to change from uphill to downhill?
So unfortunately, because of limitation of our treadmill, we did not collect downhill results. But we can probably say that step length will change with downhill. As intuitively, I would say that when walking uphill, you would increase your step length. But then when walking downhill, you would decrease your step length. And step length is controlled by foot placement. So that would change.
Great. Oops. So Yeah, your point here, which is about everyday tasks. I was just wondering if you had any sense of what falls look like, or like what instability looks like in older adults when they're walking up in slopes in everyday tasks. Has that been looked at generally in like a rehabilitation setting, or anything like what a hallmark of a fall on a slope, what might precipitate a fall on a slope in an older adult?
So there are a lot of reasons why older adults fall. So when they fall on living ground walking because of higher instability, because of like neural noises. If you are trying to make that task more difficult for the subject, especially for older populations, they may need like to generate more power to walk up a slope. So the noise can increase even more when they're trying to achieve such a task. So it's not well understood, and this is why I started with this project in the first place.
Yeah, that's really, I think it's really interesting work. And so it makes sense why you'd go down this road if there's not so much research that's been done there yet. We did have another question come in the chat. Have you thought about implementing your model in bipedal robots?
Not currently, but that's definitely something that I can think of in the future.
And okay, cool. And then last question, and then we're gonna have to wrap up the session. Do you think it's possible that foot placement strategies are impaired in older adults?
Yes, that's definitely a possibility. Especially that, healthy subjects can use foot placement to modulate their walking or to recover from deviations and perturbations and even external forces, but older adults cannot do that, and that's can definitely be affected.
Cool. And then just so you know, the person who asked that question has just suggests checking out 2 recent papers. So you might want to just open up the Q&A just to make sure you can have a look at that. So with that, I think that we'll wrap up this session. Thanks everyone for your questions, and thank you, of course, the panelists for these great talks. And we hope to see you at some future NeuroMatch sessions. Ciao.
Thank you.
To my time, it's 7. Can we, or shall we start, or wait a bit?
You can start, I would say.
Okay, no, because I see recording paused, that's why I
okay, I'll just click on recording.
Yeah, thanks.
Yeah.
Okay, cool, thanks. So, hello, everyone. Welcome to the session. I'm Ezgi. I will be your host. I'm a postdoctoral student researcher at the Justus Liebig University of Gießen. And I will introduce the speakers. We have 3 great speakers today, and the session will be about sensor processing. The talks will be on parasaccadic visual processing, odor processing in mouse models of autism, and also the use of optogenetics for microstimulation. This is an interactive session, so if you like, you can come on stage after the talks to ask your questions live. And if you want to do so, you can just let us know by writing on the question part that you would also like to come on stage. So we will make you panelists and you can interact with the speakers. Otherwise, I will ask the questions for you. You can also use the chat to tell about yourself or interact with also other attendees or speakers. So we can get started. Our first speaker is Amir Hussein Gaderi. He's a postdoctoral researcher at York University and is working with Doug Crawford. And his talk is titled Functional Brain Network for Parasaccadic Visual Processing, Evidence from High-Resolution EEG and Graph Theory Analysis. So the stage is yours.
Thank you. Hi, everyone. Thanks for attending to my presentation. Here, I want to talk about a functional brain network that is involved in pre-saccadic visual processing. And we used high-resolution EEG and graph theory analysis approach for analyzing the EEG data. So, for the several brain regions are involved in pre-saccadic perception and processing, including posterior parietal cortex, supplementary motor area, and frontal eye field. And because we have a distributed brain regions that are involved in this function, we can say that a network is involved, but nobody look at the whole network that is involved in pre-saccadic neuron, pre-saccadic perception. And here we are going to use the graph theory analysis to investigate this network. First, I should talk about the method. We had a psychophysics task. We presented one stimulus repeatedly, and after that we presented a novel stimulus, and the novel one is now we name it test stimulus. And finally we asked them to judge about the duration of the test stimulus, and in this paradigm we had two conditions. One condition is fixation task, and another condition is saccade task. Everything in saccade task is similar to fixation, but we ask participant to perform a saccade 100 milliseconds before the presentation of test stimulus. And exactly during the presentation during the saccade or pre-saccadic interval, as you can see here in the figure here, this time is known as a pre-saccadic duration. And during this interval, we analyze the EEG by source localization using the Loretta. And after that, we perform connectivity analysis to find the whole brain network. And you can see some details about the EEG and participant here. So we found this network. Actually, this is the difference between the saccade and fixation. And we found these connectivities in different frequency bands. And here, the first result, we found some brain regions that are involved significantly different in the saccade versus fixation. In the alpha band, we find inferior parietal cortex, Brodmann area number 39, and temporal pole, Brodmann area number 38, are playing the role of hubs in the network. And in the beta network, we find Brodmann area 8, that is actually the frontal eye field is involved in the saccade as the hubs. So, but other result, first we look at the clustering coefficient of the network. And clustering coefficient in a network is related to the segregation and local processing in the network. And we find that in all frequency bands, we have higher values of clustering coefficient in saccade versus fixation. The second measure that we investigated was efficiency or global efficiency, that is related to integration in the network, and same as clustering coefficient, we find increased activity for that. And then we can say that the network during the saccade is more integrated and more segregated. After that, we look at the energy of the network. Again, we found the increased value during the saccade in all frequency bands. So we can say that the network in the saccade is more synchronized in comparison to the fixation. And finally, we look at the entropy of the network. And again, we find increased activity, increased entropy of the network during the saccade. So we have more complex network in the saccade condition. So this result suggests a specific functional brain network that is involved in pre-saccadic interval with high level of integration, segregation, synchronizability, and complexity. And this brain network can prepare a neural mechanism for very fast information integration in the pre-saccadic interval. And in the network level, this mechanism can be used to fill the gap of information during saccade by merging the information from the pre and post saccadic visual fields. At the end, I should say thanks to my co-authors, Dr. John Douglas Crawford and Dr. Matthias Neymer, and all of my colleagues in Dr. Crawford's lab. And thank you for attending.
Thank you very much, Amir Hussein. It was really a very nice talk. Thanks a lot. So in the meantime, you can just post your questions, or if you want to ask, just let us know. I have one question. I mean, maybe it might be too broad, actually. But you were saying that these saccades were, so there was increased entropy, integration, segregation, synchronization, right? So what does this actually suggest?
So as you know, during the saccade, we should integrate the information from the one fixation point and another fixation point. And to this aim, brain need to integrate the information very fast. Then technically in the network level, we need very fast information processing to merge the previous data and post data. And we find this network actually has these properties. It is higher, it is more segregated, more integrated, more synchronized, and actually it is very complex because it needs to merge the information from the post and pre, and then it is very fast network, very optimized and fast network. So actually, this is the goal of this research. Yeah.
Mm-hmm. Thank you. Thanks.
I have a question. I have a question. So, so you recorded from both the left and the right hemisphere. So, is there any modulation on the parameters that you measure, like the entropy, the connectivity, the energy, depending on where the saccade is going to be, or where does it start?
So we in the psychology in the psychophysics paradigm, we had first we had vertical saccade to the left and to the right, and when we compared the saccades to the left and saccade to the right, we didn't see any significant change in the network properties, and actually the degree of the saccade was just 10 degree, but I am not sure if we have, for example, the a bigger saccade or maybe a vertical, a horizontal, a horizontal saccade. I am not sure about the other type of the saccade, but in this stage, we didn't say that there's any difference between saccade to the left or to the right.
I see, and this is on the binocular field, right? On the where you can see with both eyes.
Yeah, and all of the participants was first was fixed at the center of the screen, and then we asked them to perform a saccade to the left or to the right, as you can see here.
Okay. Oh yeah. Okay. Thank you.
So yeah, so there is a question from YouTube, and it's from Daniel Kramer. And the question is this suggesting that the brain operates with such kind of networks to solve tasks and or to make decisions, and that they get implemented by the neurons itself?
So here we are talking about, we are not talking about, for example, decision making at all. And the duration that we investigated was just in the pre-saccadic interval, before decision making, before the, for example, a further cognitive function. Here is just, this duration is related to when we are preparing to perform a saccade. It's not yet before the saccade execution. Even. And but I am not sure, maybe we can test same methodology, EEG and graph theory analysis, and we can investigate same measures to, for example, find a network that is involving decision making, and this thing is related to the EEG, and we know that EEG is related to the neural activity in a very, very big scale. And I am not sure about the result when we are looking at just a single neuron or just a small network of the neurons. Yeah, but yeah, we can technically use the same approach for different data, even fMRI or electrophysiology is recorded.
Oh, sorry, I was on mute. So I just thanked for the questions. And I would like to thank you, Amir Hussein, again, for the very nice talk. We can move on to our next speaker. Just checking. No, I'm not on mute. Yeah. Okay, cool. So our next speaker is Gonzalo Otazo. He's an assistant professor at New York Institute of Technology at the College of Osteopathic Medicine. And his talk is titled Odor Identification in Novel Olfactory Environments is Selectively Impaired in a Mouse Model of Autism. We will also post his Twitter handle and also link to the recent preprint. So the stage is yours.
Thank you very much for this kind introduction. So autism presents with problems of generalization to novel stimuli. For example, a person with autism can recognize dogs, but then but if you're presented him with a dachshund, then he would think that that dog is a cat, as described by Temple Grandin, the autism advocate. So our hypothesis is that there is a fundamental computation that is affected in autism, and we want to use to determine if this computation is also affected in mouse models of autism using olfaction. So our hypothesis is that the wild-type mice, neurotypical mice, rely on a robust algorithm to identify the target odors in novel olfactory environments, and that this computation is affected in the mouse model of autism and should not be able to identify targets in novel environments. So in order to approach this, we use a commonly known task called the CAPTCHA. So you know CAPTCHA is in the internet, the visual CAPTCHA, where you have a known target, usually some letters, and then you put some novel backgrounds and on top of it, and then you have to read the letters. And this task actually is computationally very hard. That's why we use it, because we don't know how to solve this as efficiently as the brain does. So we want to translate this into olfaction. So we're using head-fixed mice that are water deprived, and then very quickly we can train these mice to lick if there is one of the two target odors, and to refrain from licking if there is the no-go odors. And our task is a little bit more complicated than that. There is a training set where there is a contextual background, then there's limonene, and then the target appears, and based on the target, the mouse decides to lick or not. And then we can train animals to do these tasks, and over 9 days, they are doing it very well. And then one day, we are going to replace this limonene by a novel background odor, and that is our olfactory capture. So in order to quantify how hard this task is, which is one of the problems of working with olfaction, we use intrinsic optical imaging to observe the glomerular activation produced by the odors. So the backgrounds that we use with the contextual background, these 4 contextual backgrounds are actually strongly activate the glomeruli, much strongly than the targets. The limonene is like, it's sort of weak, and the novel backgrounds are intermediate between the targets and the contextual background. So to give you an idea how hard this is, these are imaging of the mixture that the mice need to classify. These are the non-go mixtures. So here, this odor includes these mixtures include the go target, and this one includes a no-go target. And they have, because the backgrounds are so strong, at the end, the mixtures, the no mixtures are very similar to the no-go mixtures. So by itself, even if you know the odors, this task is hard, but then we make it even harder to make it into a captcha by replacing the limonene by this novel odor acetal. And then now it becomes much harder. You have to, this example, for example, should classify is to the go mixtures and not to the go. So it's actually a hard task. And mice, the olfactory experts that they are, they solve this task at 77%, not as well as the known backgrounds. I mean, with the known background, the animal practice this over thousands of trials. They don't do as well, but still is pretty significant, with 50% being chance. So, so we're very happy that wild-type mice can solve this task. But as it turns out, a very simple algorithm called the nearest neighbor, using the glomeruli data from our imaging, can actually match very well the performance of the animal. So here, each dot corresponds to the performance on a novel background odor, and this nearest neighbor can be implemented using the circuitry in the olfactory system, and it just does the simplest thing that you can imagine. So you get a novel mixture that includes a novel odor, and then find the best match, and the best match is a go, then this should probably go as a go mixture. And that very simple algorithm predicts the animal behavior. Are the animals doing this very simple algorithm? We don't think so, because there are 4 contextual background odors, 4 2-go odors, 2 no-go odors. So there's a total of 60 mixtures on the training set that we use for the imaging and for the behavior. And then we reduce that by half. So the animal is going to get half of the training data. And when we do that, these algorithms, the nearest neighbor and also the linear classifier performance drop. And a group of 3 new animals that were trained with this reduced set, their performance was unaffected compared to animals. So the animals are doing some very sophisticated algorithm that can actually, with very few training data, can perform very well. So they are very good at generalizing, better than these simple algorithms we try. Is this computation affected in the mouse model of autism? So we use the CatNap2 mouse model of autism, which produces seizures and autism in humans and hyperactivity. And people have looked at these mice, and they do have a phenotype where they show reduced interest in social odors. However, these CatNap2 knockout mice are not anosmic. In fact, they are twice as fast as wild-type mice in finding food buried in bedding. So we look at the glomeruli responses using our intrinsic imaging, and they were almost identical to the wild-type mice. We look at, try to use some classifier, and performance was almost identical. So, in fact, there's no difference on the performance of these animals when they train as fast and equally well as the wild-type mice, and they do very well on the known background odor. Remember, this is very difficult, very subtle differences, but this catnap2 knockout mice have no problem on that. However, when we change to novel background odors, the performance dropped significantly. So these mice are not able to solve this task with novel backgrounds as well as the wild-type mice. So what are the possible confounders not related to the olfactory processing? One is that these catnap2 mice are hyperactive. They cover twice the distance than wild-type mice. So maybe they have problems with holding licking when you are presented with a novel background odor, and they will lick wildly. But that's not what happened. In fact, the opposite. The catnap2 mice, the type of mistake that they do is they fail to lick when there's a go odor in the presence of a novel background. So that's what they do. So it's not that they are hyperactive, or that hyperactivity is not affecting this behavior. Another possibility is they have reduced interest in the odors, so they have less time to explore. However, they have the similar phenotype. This is an head-fixed behavior. When you present a novel odor in the wild type, the mice increase the sniff rate, and the catnap2 do something similar. When they present it with a novel odor, they explore it. So the exploration seems not to be affected. What seems to be affected, we found it very interesting, is the reaction. The wild-type mice, they slow down when they're challenged with a novel background odor. And the catnap2 don't do that. So we believe is that this computation takes time, and the mice are actually, this computation requires time, and the catnap2 cannot engage in this computation. So I'll show you that wild-type mice can solve these olfactory captchas, and the catnap2 mice cannot solve this task. And I would like to thank you for your attention. This is the students that helped me. Thank you.
Thank you very much, Gonzalo, for the very nice talk. Thanks a lot. So let's check if we have any questions. For now, I don't see any questions, but I have a question. So you say that the catnap mice were twice as fast as finding food than wild type mice, correct?
Yes, yes.
Why do you think that might be the case? Just because of their hyperactivity, or?
That's a good question. We don't know why. One possibility is that they detect the odor, but then they move much faster and dig the food faster. But that's just to illustrate that these mice are not anosmic. And in fact, if you look at the periphery, their olfactory processing seems to be unaffected.
So you don't think that that might be the case?
Yeah, yeah. I mean, there's all these. People with autism, sometimes they have like abilities that exceed the neurotypical people. For example, they are very good at finding small details. And I think that this mouse model of autism might have that ability to do like this very fine discrimination, as long as it corresponds to a stimulus that they know pretty well. So the bedding, they know pretty well. The food, they know pretty well, so that they are pretty good at finding, and might be even better. We have not tested that, but this is just to illustrate that they are not actually anosmic. That there might be other mouse models of autism that have anosmia or hyposmia, and then they should not be able to do this task as well. That's why we actually choose this one, because we knew that they were pretty good at doing olfactory behaviors.
I see. So is it because that drew my attention, but maybe, I mean, I'm not that familiar with the topic. So the number of mice that were included in each condition were like 3, 4. Number of trials were high, I guess relatively high, but would that be the case, or it's like just a preliminary?
So this is for the poster. We have tested, I'm showing only like 3 of the wild type. We test only 3 mice on this task, but we tested different variation of this type. In total, we have tested over 17 mice, or almost 20 mice. and the performance is always 77, 76, doesn't change much. So it was really, respect to that distribution, these formats stand out that they draw. And we have the control tasks that they are actually engaged and behavior is fine. But of course, we would like to increase our number. We also are interested not only of focusing on the CAPNAP2 mice, autism, each mutation in autism corresponds to very few cases. So we actually are interested on testing other models of autism. And we're also interested, these are genetic models. There are also some environmental models like the valproic acid, and we're also interested in testing if they would also show this phenotype. Yeah, this is step one.
Mm-hmm. Thank you. So we have one, actually, no, two, no, just one question, right? So shall I just read the very
yes,
last
please.
one? Yeah.
Yes, please.
Okay. So it's from Hector Baez. Does increased false rejection occur primarily because mice take much less time in decision making compared to the wild type? So that, yeah.
False rejection. So I mean, the mice take the CatNap2 knockout mice, when they react very fast, faster than the wild type mice, and similar speed as when they do the task with on the known orders. So you would have expected that because they react so fast, then maybe they should produce actually false positives. But the error that they have is false negatives. So the order is there, the target is there, but they don't leak. So we don't know exactly why. One possibility is that the mice don't like drinking water in the presence of this novel odor that they might find aversive. That's a possibility. However, when the mice leak, we immediately close the odor so that the water is not going to have that. We try not to have present the odor with the water at the same time. However, we don't know if that will be enough to clear out the odor from the nostril. It seems that, and in all the behaviors that people have tried for having training mice to doing this leaking go-no-go task, head-fix behavior, in most of the cases, the errors that people see are mouse leaking for every other. So it actually was very surprising, not only in this task, but in the context of the other tasks that people have tried, that they don't leak. And that's actually, we found that very surprising. So it seems to us that either the novel odors are really aversive, although if it's really aversive, the best thing that you can do is leak, and then that stops the odor delivery. Or the other possibility is that they actually are able to detect the target odor. One speculation that we do is that maybe that's why novel odors are aversive for this mouse model of autism. Is the same way that for children, human children, going into a dark place is distressful, because you won't be able to detect what is in there. So if you enter in an environment with a novel odor, maybe these mice know that they are not going to detect if there are predators or anything, and that might find distressful. That's just an interpretation. But I tell you why, like from the point of view of the mice, how he perceived that, we don't know yet. But actually, it's a hard question to answer. Thank you.
Thank you very much. Thanks a lot also for the talk, and thanks to the panelists and attendees for the questions. So we can move on to the final talk of the session. So the final talk is from Hector Baez. Hector is is a graduate student at the University of Rochester and is working with Dr. Gomez Ramirez. His talk is titled Microstimulation and Optogenetics, a Combined Stimulation Strategy.
Great, thank you so much. So, yes, my talk is on a combined stimulation strategy, specifically to improve targeted activation of functional cortical ensembles using microstimulation and optogenetics. So, intracortical microstimulation has been used to study the functional role of brain areas and to provide sensory feedback in brain-computer interface applications. ICMS is most effective when applied to topographically organized encoding areas encoding similar features, such as in primary visual or V1. However, most feature representations in sensory systems, especially in somatosensory, are not topographically organized into columns, but instead are encoded by sparsely organized neurons. So using ICMS to target these motion ensembles may activate neighboring cells tuned for other features such as texture. An alternative approach is, of course, optogenetics, which enables us to activate or inactivate single cell at the single cell level. However, increasing intensity to cover a larger area loses the single cell resolution, and also we are increasing thermal toxicity. So the goal of this project is to reduce the ambiguity of artificial sensations by implementing a combined strategy of intracortical microstimulation and optogenetics using and to do this we're using a biologically constrained model of the SI cortex. So microstimulation and optogenetics each have distinct intensity distance relationships. Microstimulation is applied to cortical tissue through electrodes that deliver current spreading concentrically around the electrode tip. And the effective distance of excitation is proportional to the square root of the current intensity. On the left figure, neurons closest to the center of the stimulus are activated most strongly, of course, with the effect dropping off over distance. However, some neurons far from the center are still activated, likely from their axons traveling to the nearby source of stimulation or to local circuitry. And that can be as far as 1 millimeter distance from the center of the electrode. Luminous intensity originating from a beam of light, in contrast, dissipates rather quickly. And in brain slice, 1,000 milliwatts per millimeter squared in the center is sufficient to reliably activate a neuron, but this very quickly drops off. The model anatomy is anatomically organized based on the hand representation of the S1 cortex. According to Mountcastle's organization, each digit is assigned a spatial representation on our 2D plane, and neurons are spread out across 15 pad representations shown on the image, on the first two images there. The model is also comprised anatomically and functional interactions between excitatory and inhibitory cells representative of the EI balanced networks. Within those local circuits, we have a 1:4 excitatory inhibitory neuron ratio. And I have an example of this in the bottom left figure, where in this 2D representation, green dots represent motion encoding neurons, which we want to activate to induce some sort of motion percept. Blue are excitatory neurons encoding other features. Red are inhibitory, while black are axonal networks. And so we are using Poisson statistics to construct our spiking model. So the probability of spiking occurring in any time bin dt is simply r multiplied by dt. And within this simulation, we are modulating the r value. And so the way we decide if a neuron is spiking is if the neuron is spiking 50% of the time greater than normal, then we see that that neuron is activated. And our model also incorporates neural connectivity, where on the core scale, long-range projections can preferentially link cortical areas with similar responses to sensory stimuli. In this case, the similar responses encoding similar motion directions. So our combined neuromodulation approach is made possible through a transparent microelectrode array developed by our collaborators. And this array is capable of recording as well as delivering electrical current and also light stimulation to drive optogenetics. And so we used a particle swarm optimization to find the best stimulation patterns, which would selectively activate motion encoding cells. And we utilized this particle swarm algorithm to iteratively achieve these results, where a lower ratio represents a higher motion encoding selectivity. We found that microstimulation alone will always perform the worst, activating 50% more non-motion encoding cells compared to motion encoding cells. opsogenics can be used in this setting as either an excitatory opsin, or in the purple as in yellow as an excitatory opsin, or in the purple as an inhibitory opsin. And we found that the inhibitory opsin, as it currently is worked into our model, it works better than the excitatory opsin for creating a more robust artificial sensation. And yes, those are my conclusions. We were able to reproduce some in vivo data where we found that neurons closer to the source are activated much more easily, of course. But we were also able to determine that using this combined strategy, we're able to selectively activate functional sets encoding motion, encoding neurons. And we're also able to deliver these stimulation patterns with lower intensity profiles, which would result in lower toxicity for the cells. And that is all. Thank you.
Thank you, Hector, for the excellent talk. That was really interesting. So just feel free to post your questions on the Q&A, or you can also come to the stage to ask your questions. In the meantime, I can maybe ask, it might be a very naive question, of course. As I said, I mean, like, I'm really very unfamiliar with the technique. So do you think it would be possible, or maybe like as a next step, as a future step, it will be possible to use this technique and because there is efferent and reefferent feedback, to study the contribution of efferent and reefferent feedback. I guess in animals that's possible, but
I'd have to say that I'm not entirely sure. As long as using this technique, of course, we'll be able to study functional sets encoding very specific tuning, such as motion or texture, or if we were studying V1, of course, we could study orientation columns. So perhaps we could see something like this. Yes.
Intensity.
Perhaps.
So, like, if you think about the intensity of a somatosensory stimulus, maybe it might be possible.
Perhaps it's difficult for me to say for sure.
Okay. Okay.
Yes.
Yeah. I mean, again, as I said, I'm very unfamiliar with the method. So we have one other question from Devon Griggs. I hope I'm pronouncing the name correctly. Which opsins were used or would be used?
So in this case, we modeled two different opsins. One, we used a excitatory opsin and an inhibitory opsin. Specifically, they were quite forgetting the specific name of them, so I apologize on that. But functionally, the function of one is simply to silence at the single cell level whatever cells were encoding non-motion, anything that wasn't encoding motion.
Okay, thank you. Also, thanks for the question, Devon. If you have any other questions, again, panelists or attendees, you can either come to the stage or post your question at the Q&A.
I have a question for Hector.
Sure.
So I missed that part, but sometimes when you excite cortex with a current, and you recruit recurrent inhibition, and then you have the paradoxical effect that you increase the current, and then the cortex goes silent. So have you seen that, and are you always also going to model that, the recurrent inhibition?
So our plan currently is to model this in our model, but we are beginning this combined strategy in the mouse model right now. We are aware of that, but we haven't started yet, so I can't say for sure that we have seen it or not. But that is certainly a problem that we are aware of.
Thank you.
Thank you. Also, Gonzalo, for the question. So if there are no more questions, I think we can end our session. So I'd like to thank the speakers for the wonderful talks, and also attendees for their excellent questions and engagement. And also especially thanks to our backend, Reka Varya, who's been doing all the technical work. And with that, I wish you a nice day or evening.
Okay. I see we have the light came on that says that we are live and we are right on the hour, so we can go ahead and get started. Welcome everybody to one of the sessions of the final day of NeuroMatch 3.0. I'm Emily Ari-Jones. I'm a postdoc in the Giacomo lab at Stanford, and I will be your front of the house host. We have Gauri Somasakar in back of the house, who's going to be managing all of the Zoom webinar stuff. We have an amazing lineup of speakers today. Please post any of your questions in the Q&A panel. This is an interactive session, so we're kind of looking for lots of discussion. You can post them at any point and upvote questions that you think are interesting. I will ask them live for you. And everything here is public. I will post a link to YouTube in the chat, and please tweet whatever you feel like using the hashtag NMC3. So for today's speakers, we are going to be discussing new tools for studying non-human primates. We're going to learn about two methods of tracking behavior and one method of electrode implant design. So our first speaker of the day is Felipe Parodi, who is a PhD student in neuroscience at the University of Pennsylvania, working with Michael Platt and Conrad Korting on tracking the rhesus macaque in live, unrestrained social interactions. Today, he's going to be telling us about a tool to track these natural behaviors. So please take it away, Felipe.
Cool. Thanks for that, Emily. Let me go ahead and share my screen. Let me share this one to you. Okay, what do you guys see right now? Let me switch that. Do you guys see the PowerPoint?
Perfect. Yes.
Cool. Great. Yeah, thanks for that intro. As Emily said, my name is Felipe Parodi, and I am going to be talking about using pose estimation to study unrestrained social interactions in the rhesus macaque. And I'll spend the next 5 minutes talking a bit about quantifying social behavior in the macaque, what's been done so far, and what we're excited to do in the future. So to start, I want to introduce our main character, the rhesus macaque. One of the most important model organisms in neuroscience and other fields that are impressive for a variety of reasons. So one of the ways to gauge an animal's success is by using the criteria of population size and geographic distribution. And when we look at this in the rhesus macaque, it turns out that this animal is just as successful as humans. And this is largely due to their social nature. And for social primates like humans and macaques, their success largely depends upon their ability to cooperate, compete, and emotionally bookkeep. That is, track and remember previous social interactions. And this is significant because social deficits in both humans and macaques is associated with chronic deleterious impacts on health and well-being, making their study a priority. And so how can we study social behavior in the rhesus macaque? A tried and true method to employ techniques is to employ techniques such as focal animal sampling to manually inventory the behaviors of a single animal to construct what is called an ethogram, which is exciting and useful, but it comes with a number of issues. Conducting ethograms is time and labor intensive, and it's pretty vulnerable to experimenter bias. Now in the lab, on the other hand, studies in humans and macaques alike have been restricted to highly controlled settings, largely for practical reasons. These studies typically involve restraining a monkey, as we can see here, so that it can interact with a computer, often completing economic games. And what people have done is record from an electrode and associate neural signals with mathematically tractable variables modeled through game theory. And this research is exciting and valuable, but as Yale Niv recently put it, we tend to overestimate neural data while largely underestimating behavior. You see, measuring behavior in highly controlled lab settings is a bit inorganic, and I think intuitively most of us feel this way. And so the big issue is that we simply don't know the extent to which the specific neural processes identified through these approaches contribute to real-world social interactions. And so fortunately, recent advances in deep learning-based pose estimation, like Deep Lab Cut, have given us this rich opportunity to measure animals and their naturalistic social interactions. So to train deep neural networks, a big challenge is simply not having enough data. However, we can now leverage available macaque data sets, such as OpenMonkey Pose and Macaque Pose, to train a network to track rhesus macaques moving through space and time. Okay, so how exactly do we track poses? Well, a typical framework tends to involve recording some video, extracting frames to label features of interest, and training a network to detect these features. We then apply a connected skeleton and use this model to analyze new and diverse interactions. And down the road, we can conduct further analyses to look at three-dimensional pose estimation. On the right here, we have a picture of some recordings I was working with using Deep Lab Cut. We have a frame of one macaque grooming another. The colored dots represent our features of interest, which include left ankle, left knee, hip, among other joints. And we have the XY plot of the feature trajectories. Now, this talk was really just intended to give you some ideas of the projects I'll be working on. However, they're not exactly without their challenges. And so some challenges we face include 3D pose estimation with a nice asterisk next to it, as OpenMonkey Studio, published last month, has managed a way around this with an interesting but complex setup. Tracking in real time with another asterisk next to it as our third presentation today. I believe we'll talk more about this. And proper segmentation is yet another issue, which is simply the partitioning of an image into segments used to locate specific objects or boundaries. And finally, the embedding problem, or the ability for a computer to make sense of a given pose. See, poses tend to reflect context-dependent strategies, and with an animal that has as many degrees of freedom as a macaque, this isn't an easy task. So finally, some long-term questions we hope to pursue include taking neural recordings during naturalistic interactions between a pair of monkeys, like in the grooming image we just saw, or in group settings. And this would obviously be exciting to further characterize the neural processes underlying the macaque's social hierarchy, for example. Another approach is to the fully automated quantification of social interactions. Now, we envision, or rather idealize, a high throughput pipeline which would be fed recordings of macaques interacting and would pop out an objective, accurate ethogram. And there are a couple of packages out there that do this. And finally, characterizing social brain circuits, really making sense of what processes and agents drive social behavior and what exactly happens in disorders where social impairment is common, such as autism and schizophrenia, which would be exciting. And so with that, thanks for listening. I'd like to thank the K-Lab, the Platt Labs, and all of the NeuroMatch organizers, including Emily and Gauri.
Thank you. Don't usually get thanked on the slides. That's very nice. All right, so while we wait for some questions for the audience to come in, I had a few questions. So first was relating to the pair interactions. How do you track two entities and make sure that they are treated as like separate and their identities are kept with them throughout the tracking, even if they are like, say, you know, passing in front of each other or over each other if you're doing like 3D tracking?
Yeah, so you're referring to like pose tracking two separate individuals?
Yes. Yeah.
Yeah, and so Deep Lab Cut actually came out with like a multi-animal, like Deep Lab Cut multi-animal scenario. And so there, what you could do is you could identify two different individuals and apply a skeleton for each, and it would use like bottom-up image processing to like once it finds the left ankle or the left like a specific joint of one individual, it will connect the rest of the skeleton. But that's one of the issues I encountered. I mean, like these past 2 months, I've been working on it, right? The left ankle of one macaque tends to get confused for the right hand of another. Yeah.
Thank you. All right, we have questions coming in. The first one that I saw was from Melissa Franch from the Jorkboy Lab, who has two questions. First is, how many cameras are you using? And second is, have you attempted to quantify behavior that defines social hierarchy between pair-housed animals in captivity? And if so, do any behaviors stand out?
Yeah, so so the camera question, the answer is like right now I've simply been working with 6 cameras. The goal is to get my hands on a larger number of cameras, ideally like in the 30s, about 32 cameras, and these would be 32 machine vision cameras. And so like Open Monkey Studio, the Hayden and Zimmerman labs, and at U Minnesota, they're working with about 62 cameras, I think, with an entire setup to triangulate their 2D poses. The second question, have I attempted to quantify behavior that defines social hierarchy? No, and that's actually one of the things I'd like to do moving forward, right? And so I'm particularly interested in social cognition and decision-making, and I don't exactly know how I would go about that just yet, but I appreciate the question.
Great, thank you. And then we have another one from Jesse Fu, who asks, what is the maximum number of animals that can be tracked with the current tools with good resolution?
Yeah. That's a good question that I don't think I know the answer to. So like any animal in general, I know there's a lot of work. So like humans, like pose estimation in humans is phenomenal, right? We can track entire groups of humans with just about no problem. And I think mice, which I believe Gary will talk about later, like mice work is also, we can track, I don't know, up to like 3, maybe 4, or even more at a time. And as for macaques, it's right now it's only really at like 1 to 2 macaques, and you saw like the 2 data sets that just came out that were on a slide earlier. I mean, they just came out. One of them is still a preprint; it hasn't been published yet.
Very cool. And Zerna, do you want to ask your question? You can just unmute.
Yeah, sure. Hi. So my question is, how large is the area that you're covering right now with your 6 cameras, and and why do you think that the 32 cameras would be necessary? Yeah,
okay. Good question. So the area right now, I'd say it's about, so it's a hexagon, and I'd say it's about 10 feet across. Think like panel to panel. And height, it's about 10 feet tall as well. Now, so like why did I throw out the number 32 cameras? So like I met with Jan Zimmerman a while ago, and that's kind of the number he gave me to start gauging like when we're working with a specific number of cameras to do 3D pose tracking. This is kind of like the number of cameras that will help you out, right? With a specific model organism that has as many degrees of freedom as the rhesus macaque, right? Things are a bit different when you're looking at the side view of a horse, for example, or really just a primate face. Yeah. These are a lot of questions.
Yeah, I have another one, which is about tracking. And this is also a question I'm going to ask to Gary as well when we get to his talk, is tracking in disordered contexts. So making sure that tracking that works for normal, healthy movement is also going to work for tracking like a Parkinson's model or an MS model or an ALS model.
Oh, I'm sorry. I don't think I caught the question exactly.
Oh, sorry. So the question is I kind of would envision that there would be some challenges with making sure that you're comparing apples to apples. I could envision a scenario where you need to actually label frames from a motor disorder model and the controls separately, and that then you might not be tracking things with the same fidelity.
Yeah.
I mean, do you get that impression from, I don't know the behind the scenes about the algorithms, but do you get that impression that you would need to do that, and would that lead to some kind of differences in the labels that you would get out from those two data sets?
No, that's a great question. And I know, at least I know, in humans, to look at impairments in gait and any other specific like skeletal muscle disorders, right? They do include a very diverse data set that includes that would include these specific an impaired gait, for example. And I imagine if we're looking at like a rhesus macaque, I don't know what a specific altered behavior would look like. But yeah, I imagine we should include it in the data set. And if anyone's opinion differs, I'd love to hear that.
Maybe Gary can share after his talk. Okay, well, that has led us pretty much right up to the 15. So thank you everybody for all of those questions. If you have any more, you can post them to the chat or to the Q&A, and Felipe can answer them there. Thank you again, Felipe, for that great talk and lots of great answers. Our second speaker is Zerna Ahmed, who's a PhD student from the Sensory Motor Group run by Alexander Gale at the German Primate Center. And today she's going to tell us about a protocol for extracting skulls and designing implants for those skulls.
Thanks a lot. Can you see my slides?
Yes.
Okay, perfect. Yeah, so welcome everyone. So I will dive in right away. So today I'm going to present our universal guide for custom fitting implants for continuous and discontinuous skulls. So in primate neurobiology, chronically implanted devices such as head posts are used for head stabilization or chambers to gain access to the brain to study its activity. And such implants are skull mounted and made from hard, durable materials, as we were discussing before already. So they need to be very well fitting because you want to avoid infections and especially tissue growth. So recent approaches actually started to design those implants virtually before even manufacturing and to virtually match them to the skull surface. However, with our guide, we want to introduce a method that even expands this functionality and usability and is at the same time very well suited also for non-cut experts. So our guide is not only suitable for even skull surfaces, but it also is very well usable for skulls with special conditions, like, for example, if they have some holes in there. It additionally is it's already using the so-called virtual bending and virtual cutting approaches, to which I will come later, but we are also introducing the virtual bending approach. And the final design can be used for 3D printing or CNC milling, which allows you to use a variety of materials. So before we dive into the specific steps, I just want to very briefly give you an overview of our technical specifications. So in our guide, actually, we are also explaining how to extract brain and skull models from MRI or CT scans by using 3D Slicer. However, now I want to focus on the implant designing procedure. So the raw designing can be actually done in any CAD software of your choice. So in our guide later, we are actually explaining how to use this by using Fusion, but you can also use any other software that you want to use. And the fitting is then done in Rhinoceros 5. And actually, this is also a program, also a CAD program, with which you can also already design the raw implant. And then the manufacturing was done by using either CNC milling or 3D printing, but then again, I will come back to this later. So coming back to the first point I made before, so as you can see here on the left, we have a skull of a monkey from our lab, and his skull contains clearly several holes, and that was due to a prior infection. However, the animal was then completely healthy, and therefore we wanted to implant this animal with a head post for head stabilization and also with a broad expanded chamber for wireless recordings. So details about the wireless recordings itself, and especially about the wireless chamber design, can be found in the paper of a colleague of mine by Michael Berger et al., who published actually this year. I just mainly and briefly want to focus on the implant itself. So as you can see on the upper bar, we were not only able to match the skull surface with our head pose, but we could also take this hole actually into consideration while designing, and then, for example, giving an extra protection by covering this hole with an additional leg. And coming back to the lower part, we can see that that we managed to even customize the implant that is quite extended, like it's covering 2/3 of the skull surface itself, and still doing our wireless recordings. So, sorry, yes. So all of the recently published matters make use of what we call, or what we refer to as now, virtual cutting approach, which means that you design your raw implant and then you fit it to your skull surface by subtracting the skull surface from your raw implant. And this method works actually very well as long as there is no need to maintain the constant bottom thickness. However, maintaining this bottom thickness can be sometimes very useful. For example, if you are limited in the screw length and you do not want to insert a very broad range of screw lengths, for example, ranging from 5 to 10 millimeters. So for this, we also introduce on top of it the virtual bending approach. For just to briefly describe because of time reasons. So what we do here is we project the 3D extracted skull surface to a 2D reference surface. And then on this, we design our implant. And then this 2D flat implant is then reprojected into the 3D space, which basically allows you to bend this flat implant around the 3D skull surface. And with this approach, of course, you can also combine your implants and design implants that make use of the virtual bending as well as the virtual cutting. And then in the end, of course, again, the last point that with this, our final file design allows you to either use CNC milling, which is very useful for PEEK, for example, or create implants in PEEK, and also for 3D printing. And here you can, of course, use different type of 3D printing. So for example, we use the 3D laser sintering printers to create our titanium head pose. And we also use the more commonly used fused disposition modeling for PLL or nylon. And so with this, the 5 minutes are over, and I want to summarize that our guide is actually suitable not just for smooth skull surfaces, but it's also very useful for special conditions like having holes in your skull. We are using the virtual cutting method, but on top of it, we are also adding the virtual bending method, which can be quite useful in some specific cases. And in the end, we are able to 3D print or even CNC mill those virtually designed files, which gives it a more flexibility, more broad flexibility in terms of the materials. And most importantly, we are providing actually a step-by-step tutorial, including video tutorial, that is very well suitable also for non-experienced cut users. So in theory, anyone can design it. And with this coming to an end, so it's not submitted yet, but it's very close to submission. However, please don't be hesitant and don't wait for it. Like, I will be very happy to provide the tutorial. And also, if you have any question, just write us an email. Here you can also find the GitHub page. And specifically, if you use the tutorial, we will be happy about any type of feedback that you can give us. And with that, that's it. Thanks.
Thank you so much, Zerna. While we wait for some questions from the audience, I have some questions of my own. So I come from the land of mouse skulls, so I'm less familiar with primate skulls. So kind of a naive question, how much variation do you have between, let's say, an average primate skull from primate to primate? This obviously was a unique case where you had pieces of the skull missing. But how much of a structural benefit do you get from getting to custom bend and cut pieces to exactly fit the skull versus putting the same piece on each time?
Yeah, sure. So for sure, if you would get let's say a commercially available bended standardized implant, which doesn't exist like this, but even if you would if you take a flat one and you match it to one type of skull surface, it will in 99% of the cases, I would say not fit exactly to the other one. So the skull surfaces are actually quite different. And you really want to get as much of a close fit as you can, because otherwise we have a lot of problems with tissue ingrowth and then also infection getting inside and then basically rejecting this implant.
As a kind of related question, the models you were showing me reminded me that I assume that there's some clinically used software for designing custom, say like plates, let's say a plate for a skull to be as kind of close to this as possible. Were you able to draw from those or take any inspiration from those, or are those just completely different or closed source?
Yeah, so if you're referring to like the general concept, so the general concept are for sure inspired by like the titanium implants, for example, are for sure inspired by the human medical parts. But if it's about like the filling up the gaps, like you have this special condition where the skull has a hole and you want to fill up. So in the plastic surgery, there are approaches, but they're actually limited as far as I know, as far as I'm aware, because they are mostly using the mirroring. So that means like you have a hole on the one side, they just mirror the whole skull and then subtract from each other and then you get the plates. So in our case, for example, the holes were not divided symmetrically, so it would already create a problem. So in that sense, it's a new approach.
Interesting. So do you think that that means that this kind of approach could be extended clinically at some point?
I can imagine it can be useful, but however, I'm not sure how many cases you have where this is really like necessary. Like how many cases will you have? I guess it's also a very special case there.
I was thinking partly in terms of kind of like bone structural repair, but also getting into either you know, like implanted DBS things where you need to put something, you know, external to the skull, but presumably you'd want to have it as flush and perfect to the skull as possible. Or, you know, getting into like more implant territory. That I imagine it would be helpful to actually match it to the skull as is.
Sure.
All right, we have another question from Melissa French. Thank you, Melissa. What is the age of the animals with these implants / hardware? Have you implanted or have you experimented with implants or hardware in young macaques less than 5 years old? Your work reminds me of the custom head posts offered by Rogue Research. They use animals' recent MRI scans to determine the proper fit.
Yeah, so exactly. So, I mean, we are also using the recent MRI scans and CT scans. And we do not have any, or at least I do not have any experience with macaques younger than 5, because usually, like in our lab, we start, I would say, around 6, 7. Like 7 is more the starting age, like the age for implanting. And then the first question, sorry, was about the...
Yes, the first question was about age. It was a two-part question. What are the age of the animals that you're implanting?
Yeah, so they were ranging actually from 7 to, as far as I remember, 10. So they were quite young, yes.
Yeah, I think that answers the question. Does anyone else have, any panelists or anyone else have questions?
I've got one. So what kinds of implants are you guys using this for? And is having this like customized implant, is it especially important for some kinds of implants or like certain kinds of methods of recording like versus other methods?
Yeah, so I think there we have different cases. So first of all, the head post, the titanium head post that you use for head stabilization is for sure, like it's one of the standard implants that we have. And that's something you like usually make sure that it fits very well, because I mean, it's titanium and it should fit very well, basically. The other chambers that we, especially with the design that we have, that's what I wanted to say, sorry. So like there are different approaches. You can have also head posts that just have basically an MRI pole, and then you just put cement on it and then it's kind of adapted to the skull. But with our method that we have with the long legs, you really want to make sure that it fits as flush as possible. Then, of course, then we also have the case of the wireless recordings, which is a super special case in the sense that this type of implant was not existing before. So you have your arrays, you have your array connectors, and you want to really of course, adapt this very specifically. So for these type of recordings, you for sure need this as well. With the single cell, with the single electrode recordings that I was showing before, just with the round chamber and then the legs, there the legs we put there, but there are also approaches where you can just basically use the cylinder itself and then just put cement around it again. So it's like, depends on which type of implant you're using.
Thank you.
All right, thank you so much, Serna. Thank you everybody for your wonderful questions. We're now going to move into our final speaker. So now we're going to go from monkeys to software that can score behavior for all animals, Deep Lab Cut. So our third speaker is Gary Kane, who's a postdoc in the Scott Lab at Boston University. He got his PhD from Princeton in 2018, was a postdoc in Mackenzie Mathis's lab at the Roland Institute at Harvard from 2018 to 2020, and joined Ben Scott's lab at Boston as a postdoc this summer. His research in Ben's lab is focused on the neural mechanisms of decision-making. All right, take it away. You're muted. You're still muted.
Sorry.
There you go. Now we got it.
Thanks. Okay, well, I'll dive right in for the sake of time, and thank you so much for the introduction, Emily. Okay, so deep learning-based pose estimation tools like Deep Lab Cut have recently made it possible to automatically track a wide variety of animal behaviors with a high degree of accuracy. But to understand the cognitive and neural basis of behavior, neuroscientists need tools to manipulate an animal's environment or neural activity in relation to behaviors that are being tracked. So we want to provide closed loop feedback based on an animal's posture in real time. And to accomplish this goal using the Deep Lab Cut toolbox, experimenters need to first feed images from a camera to a Deep Lab Cut network, and we want to be able to estimate the pose fast. We also are going to need to perform some post-processing steps on the Deep Lab Cut estimated pose. So this might include sending or receiving commands from an external device. So we might want to turn on an optogenetics laser based off of what Deep Lab Cut outputs are telling us. And another thing we might want to do would be to predict the animal's future posture to counteract for delays. So we want to be able to provide feedback at exactly the time the animal is exhibiting our behavior of interest. And so we might want to predict what the animal is going to be doing a little bit into the future to deal with the delay from actually estimating the pose in Deep Lab Cut. So for this talk, I'm going to introduce some new software tools that will hopefully make this process simple for everybody. So in our workflow, the first step is to export a trained Deep Lab Cut model using the new model export tool, which is in the main Deep Lab Cut package. And next, to run a real-time Deep Lab Cut experiment, this network must be loaded and prepared to analyze images. And so we've written two software packages that will do this. The first is a simple Python package called DLC Live, which is just designed for this purpose only. So it just loads networks and analyzes images. And the second is a new Bonsai plugin that allows users to load a Deep Lab Cut network and analyze images using a camera feed from within Bonsai. So importantly, within both of these tools, we provide convenient methods to downsize or crop images for faster performance. So Deep Lab Cut is much faster with smaller images. And so these methods allow you to record video with large images so you can save that extra detail for offline analysis, but you get the speed boost from analyzing smaller ones. And then finally, we need a method for feeding images from the camera straight to Deep Lab Cut. So Bonsai already takes care of this part, but we've also developed two additional packages that use DLC Live for real-time pose estimation. First, a standalone GUI, which we're calling the DLC Live GUI. And we've also integrated DLC Live into the behavioral control software Autopilot. This is a relatively new software package that leverages some very cheap hardware to run operant conditioning behavior across kind of a whole room of boxes. And then lastly, Deep Lab Cut Live was designed to make it really easy to integrate this functionality into new software packages. So it should only take a few lines of code to add real-time Deep Lab Cut to your software applications. So really quickly, just wanted to point out that we've tested this Deep Lab Cut Live package on a variety of systems. So to see the complete results, please check out our preprint on bioRxiv. But generally, if you're looking to optimize performance, then we highly recommend using NVIDIA GPUs, such as the Titan RTX. If you're trying to run this on a tighter budget, then we can recommend these NVIDIA Jetson machine learning focused embedded systems. They achieve pretty good performance, so it should be fast enough for most applications. And then depending on how sensitive your application is, you might be able to even get by with just a conventional laptop. So we've tested it on a couple of different systems with just CPUs. And so once we have the animal's posture in real time, we need to identify the behaviors of interest and control an animal's environment or stimulate or inhibit neural activity. And so using this DLC Live package, this is easily achieved using a DLC Live processor. And so this serves as just a callback function. So every time Deep Lab Cut estimates the pose of an animal, the processor is run to perform any computations or operations you desire, and there are few to no constraints here. And so since we developed this software during the pandemic with little to no access to our usual lab resources, I'm going to demonstrate this with a little experiment on my lovely research assistant, Izzy. So Izzy loves to jump up for treats. And so what we're going to do is we're going to turn on a little LED in the corner here whenever she jumps up. And so I want to point out that it's really simple. It only takes about 15 or so lines of code to implement a processor to do this. And so once we have the animal's posture in real time, I'm sorry. So you can see pretty clearly here that the system will reliably turn on the LED, but we have one small issue is that no matter how fast we can run this experiment or how fast we can estimate the pose, we're always going to run into some delays between when the animal is exhibiting behavior and when we can actually send a control signal. And so to counteract these delays, we can try to predict the future pose of the animal. And so here we're going to provide a demonstration that uses a Kalman filter to estimate the position, velocity, and acceleration of all tracked body parts in real time. And we're going to use this information to predict where each body part will be. And so again, I want to point out that this can be implemented with only a few lines of code, and the base Kalman filter is provided here on GitHub in the DLC Live package. And so without forward prediction, you can see here that we were about 70 milliseconds, it took us about 70 milliseconds to turn on the LED from the first frame in which Izzy's paws were above the level of her shoulder blades. But when we use this forward prediction module, you can see that we're much, much closer to turning on or off the LED right around the time that she was actually exhibiting this behavior. And so kind of that's the gist of what these new software tools can offer. And so in closing, I'd just like to thank Mackenzie and Alexander Mathis's labs at EPFL, and in particular, Sebastian and Jesse in Mackenzie's lab, and then also these institutions for providing their support for this work, and also Emily and the NeuroMatch conference. And so now I can open up for questions and discussion.
All right, thank you, Gary. That is the first NeuroMatch talk I have seen that featured a dog. And so I think my first question is, can we just see more videos of your dog? And while we're waiting for audience questions, I'll ask the same question that I asked Felipe, which is, what is your impression of having to score behavior from motor impairments? Like, do you think that you could, for instance, just label frames of control animals and then still be able to capture those, same points on a, let's say, Parkinson's animal model or Parkinson's human or anything like that? Or do you think you would actually need to train individually on those two different kind of motor types?
So I think it's really going to depend on what these individual frames look like. So Deep Lab Cut is choosing these labels based off of like the single image that you provided. So it's only using basically like whatever statistics it can pull out from that individual frame. And so if like the individual images, if kind of the configuration of limbs and the positions of the body are relatively similar between the videos you're taking of a healthy or control animal versus like a motor impaired animal, then it should work just fine. But if your motor impaired animals exhibit some weird kinds of movements, so like for example, when training this network to track Izzy, I had to label a ton of frames to get this to work successfully because there was just really huge variety in the frames. So like have to label kind of like the entirety of a jump sequence, and I'm going to have to label that jump sequence in all different parts of the room, because if Izzy's facing in a different direction or something, then the configuration of these limbs is different. And so it's kind of a novel image for the blab cut. And so I think it's kind of like you want to make sure that you're labeling like one image from kind of the whole complexity of the behaviors that you're going to capture.
Okay, that makes sense. Yeah, I'm hoping to see deep lab cut used on, to make more kind of automated behavioral phenotyping. Like I have scoring for things like some scoring for motor impairment. Outside of things like, gripping tasks is largely just kind of scores, and it can be somewhat subjective. So I'm excited to see kind of the capability of deep lab cut to track animals through kind of motor impairments that are unusual, especially seizures. Being able to classify seizures behaviorally would be really nice, but seems also like it would be quite a challenge once you get into like full tonic-clonic seizure range, and the possible position of any part of an animal is literally anywhere in the video camera space.
Yeah, I mean, you wouldn't have to label like one of absolutely everything, but you definitely, like, you're going to need to get like a good range of things. And so deep lab cut also provides some tools. So you can label a first set of images, train the network, and then you can easily pick out the ones that deep lab cut is failing on. So it provides kind of this confidence measure that will tell you like, I have no idea, like kind of, where so you can kind of pick out these low confidence frames where it either like isn't sure where a body part is, or it thinks that the body part is missing from the video. And so it's usually easy to identify where it's failing and to fix those specific parts.
Great. All right, we have another question from our favorite question asker, Melissa Franch. Thank you, Melissa, for all of these wonderful questions. She says, thank you, Gary, this is great. When you label different parts of the animal at different orientations, you keep the label the same, correct? Meaning there is always one label for left paw, but you will train the network on the left paw for many orientations?
Correct. So if, for example, I'll just keep talking about Izzy, my favorite subject. So in like this image right here, like you can see, so like the right paw is further from the camera at this point, but if Izzy was turned around in the opposite direction, then the right paw would be kind of the closer limb. And so that's always going to be the case, is always going to label the body parts for exactly what they are, and you can train them. It's even possible to train one deep lab cut network on multiple camera views. And so like because it's just taking the raw image and just spitting out labels based off what it has learned from that raw image, it doesn't even necessarily have to be the same camera view.
Neat. I actually had one question about the part you mentioned at the end, the Kalman filter. Could you go into a bit of detail about that to help out those of us like me who like kind of have an idea what a Kalman filter is, but not quite?
Yes. So I think the most important part of it is really just like for the forward prediction aspect is really just measuring the position, velocity, and acceleration. And so we just do that by taking, so like the position is what deep lab cut spits out as like the position on the latest frame. The velocity is the difference between this frame and the frame before, and then the acceleration is the difference between the velocity measured on this frame and the velocity measured on the previous frame. So once we have those three values, then it's a quadratic approximation. So we're literally just saying, so we're using this acceleration and velocity and just kind of extrapolating forward to predict like where do we think it's going to be based off of like its current position, its rate of change, and the change in the rate of change. So the Kalman filter aspect is just like a small trick to eliminate noise. So if you just take those raw values, then especially in the derivative, so when you're calculating velocity and acceleration, any noise that's in the position, which is like, so if you have noise in the position, it's not too big of a deal. but the noise in the position can get really amplified. So if you have like, if the actual position of the limb has stayed the same, but because of just, you know, random noise, it thinks that it moved down quite a bit, then when you make your forward prediction, that noise is going to dominate the prediction, because it's like the model is now going to think that like it's kind of moving, its velocity is moving downward, but it's really staying the same. And so what the Kalman filter does is just like a technique for real-time Bayesian filtering. So it just uses kind of the expected uncertainty or like some level of noise that it expects in these measurements to get a better estimate of position, velocity, and acceleration, and therefore giving you a better prediction.
Thank you. All right, while we had that answer, we have three more questions that just came in rapid succession. So let's see if we can get through all of them in like 4 minutes. Okay, first one from Joseph Lushinger. I hope I'm not butchering that too badly. Great talk. For more complex behaviors, there are a number of options for ethogram identification. Can you speak to how/if ethograms could be incorporated into DLC Live?
Yeah, so just for a quick answer here, the processor object is super flexible. So all it has a function called process, which just takes in a deep lab cut pose. And so you can implement whatever your ethogram algorithm is. It could be anything. It could even be another deep neural network. And you can use that pose, feed it into like your whatever algorithm you're using for your ethogram, and then you can figure out like which behavioral state the animal is in, and then do some sort of triggering or control external hardware based off of that. And so because like you can also save a history of poses, so that's kind of how we implement the Kalman filter, is we save like the last couple of poses, and so you can just implement it solely there. And so just it shouldn't be too difficult as long as you have that code already written.
Great. And then we have one from Alejandra Regla Vargas, who says, can you speak a bit more on sub-zero latency feedback? I'm not sure I've wrapped my head around what that means.
So I guess it's almost like a little bit of a joke. So the goal is to have zero latency feedback. You want to be triggering something exactly when the animal is exhibiting behavior, but there's a little bit of noise there. So, and also this experiment, we handicapped ourselves by using like a $20 webcam and very cheap hardware and things. And so we weren't super precise. And so when we were shooting to get zero latency feedback, sometimes we were a little bit early. So we were actually like turning off the light 13 milliseconds on average before Izzy completed her jump sequence. And so that was, I think we could get a lot more accurate if we had just used nicer hardware, or if we had a behavior that was a little bit easier to train with a faster network.
All right, and then we have one last question from Melissa again, who asks, do you have any experience using deep lab cut to track moving animals from a camera that is not moving? So for instance, if you strapped a GoPro to Izzy, and then you wanted to track another dog that Izzy was playing with.
I don't have experience with this, but there is no theoretical reason why it would fail. I mean, some things to consider are motion blur. So if you have a camera that's moving on a dog, then you're going to need a pretty low exposure to make sure that there's not motion blur so you can get good images. But otherwise, deep lab cut should be able to be trained to track whatever you train it to track. I think that's the biggest thing, is like whatever training images you give to deep lab cut, that's what it's going to learn how to do. So as long as you give it the appropriate training images, then it should be able to learn to track it.
Neat. All right, thank you so much, Gary, and thank you everybody in the audience for so many wonderful questions. I think we are at time to end. So I wanted to thank all of our speakers one last time, and thank everyone who asked questions, and thank Gary for operating on the back end. And we will see you all in the next session. The very last speaker session of NeuroMatch is next session.